and also appointed as a scientific authority in coaching national researchers and hosting the inauguration of Indonesian research professors. To support world-class research activities, LIPI managed 5 botanic gardens as a center for ex situ plant conservation, 3 museums that have the biggest collection of flora and fauna specimens in Southeast Asia, and regional level and Indonesian natural history. Baruna
Bulan, izin kita admit itu ya, serta ya sudah masuk.
Ya tadi memang putus-putus apa memang diputus?
Emang diputus.
Oh oke. Siap-siap ya teman-teman. Bismillahirrahmanirrahim.
Yep. Can you hear me?
Yes, good morning, Professor Sil.
Hello, good morning, Professor Say.
Sorry, I was no sound.
Hi.
Can you hear me?
Try? Yes, of course, we can hear you very clearly.
Is it clear, or should I use my microphone later?
No, it's quite clear actually, but it's quite dark, I guess, because.
Oh yeah, I will close the window.
Yeah. Let me check. Sorry, thank you.
Okay, that must be better.
Udah.
Yes, that's better.
I can see you clearly now. How are you? It's very long
fine.
time.
Okay,
I last met you on 2010. Do you want to try your presentation?
Oh, what's happening to my?
What's happening with your camera?
I had that before. I don't know if it's my computer or the connection. Is my sound still good?
Yes, we can still hear you.
Wait, maybe I will restart my computer and try again. Okay?
Okay. We'll wait for you.
Yeah, bye.
Bye.
Seems to work this time.
Hello, Professor Sil. Did you join us already?
Yes, I think it works now.
Okay, thank you.
Oh, I saw my video doing funny things. Yeah. But when I share my screen, let's see if that works. Is that okay if I try?
Yes, but please consider that we already accept some participants, so
They can see it.
yes, they can see it. If you want to present some surprise, then it won't be it won't be a surprise anymore because some participants already attending here. And probably the video is again.
Yeah, I see it. It's not good. I don't know why that's happening.
So did you need us to share the screen for you?
Wait, let me, because I think when I share the screen, you should be able to see it. Let me just try.
Okay. Mas, bisa dibantu nggak?
I cannot share at the moment, I think. Because another participant is sharing. Okay.
Yes, just wait a minute. Please be patient. Wait a minute.
No worry.
Jangan di-mute kan? Hello, Professor Sil. Can you try now to share your screen?
Okay. Can you see that?
Yes, we can see that. Biodiversity conservation in Southeast Asia, right?
Yep. So it's just my video doing funny, right?
Yes. Yes.
Okay, yeah. I think something with the driver of the camera, because this has happened before.
Oh,
Let's see. Oh, can I try? I don't know how this works when I do full screen.
yes. Yes, you can. Of course, you can try it.
Oh, how does that work? Oh, yeah. Okay. Fine.
Yes.
That looks nice.
Mhm.
Okay, so everything works except my video.
Unfortunately.
But I can try to switch it on every now and then.
Yes.
How do I do that?
Just
That's the wrong
press
one.
the page down button also can work.
Okay. Right. Okay, so I stop sharing the screen, right?
Okay. Yes. Thank you very much. So we still have some time before your session.
Yep.
Yes.
I'll
we
just
still
wait.
have. Yeah. You. Oh, thank you very much.
Oh, so Indonesia still has the virus.
Of course. How about in Brunei?
Yeah, we got rid of it in April, so it's completely free here.
Really?
Yeah.
Wow.
We're lucky.
Because yeah, you are lucky living in Brunei. But in Indonesia, the case is still increasing because we have some holiday, and I guess people is already had stress, so they want to take a holiday. They didn't. They insist to meet their family and something like that, and then there's new cluster with the family, with meetings
Yeah.
also, and in the
Same things happening in Europe.
tourist area. Yeah. Yes. So for a whole year, you stay in Brunei, didn't go anywhere.
Oh, yeah, that's the problem. We can't leave the country. Well, we can, but when we come back, we have to go into quarantine for 2 weeks. Yeah. But Brunei is a small country, so it's more easy to control who goes in and out than a
Yes,
country
I can
like
imagine
Indonesia.
that. Yeah. How about the vaccine development in Brunei? They're
Oh,
not doing
not yet.
Not yet.
I haven't heard about that yet.
Oh, yeah, because it's already clean in Brunei, right? So is it possible for the foreigner to come to Brunei?
Yeah, I can still come, but they will put you in a hotel room for 2 weeks, and you cannot leave the room.
Oh.
Before they let you out.
Okay.
So almost no one is traveling anymore. Everybody stays in the country.
Yeah, that's more safe for now. Also, welcome to all the participants. Professor Slick, I would like to introduce you. We have the head of Research Center for Conservation, or previously the head of Bogor Botanical Garden. There is a Professor Dr. Hendrian. Hendrian, would you like to say something?
I know
Yeah,
Hendrian.
of course. Pak Hendrian, would you like to?
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Hello.
Hello, Ferry. Long time no see. Yeah? I heard that you're in Brunei now.
Yep. Yeah, I want to visit Indonesia, but it's not so easy to leave Brunei at the moment because of the quarantine.
You're very welcome. A little reunion here. You would like to come?
Let's wait until the vaccine is there.
Yes. The next Flora Malaysiana will be a good chance also to gather with old friends.
Yep. At a very nice place. So.
We have also Director of Research Center for Biology, Dr. Atit Kanti. Ibu Atit, are you there already? Did you know her?
Yeah, I don't know for sure.
Yeah. she just become our new director recently. She's a
okay.
microbiologist, so maybe you didn't correspond with her. So, but it is a nice chance to meet her here also. We have also our director of Research Center for Biomaterial, Dr. Iman Hidayat, already here. I guess, yeah, but he's still on the way, so maybe.
Okay. Yeah, I was a bit early.
No,
I
we
wanted to make.
are happy to meet you early.
I want to make sure that everything worked.
Yes, of course.
So maybe now I just wait until I think when it's 9 at your place, right?
Di Uzbekistan ada padang terbuka dan berdebu. Aneh, aku jadi ingat pada umbu. Rinduku pada Sumba adalah rindu padang-padang terbuka, di mana matahari membusur api di atas sana. Rinduku pada Sumba adalah rindu peternak perjaka, bila mana peluh dan tenaga tanpa dihitung harga. Tanah rumput, topi rumput dan jerami bekas rumput, kleneng genta, ringkik kuda dan teriakan gembala. Berdirilah di pesisir, matahari akan terbit dari laut dan angin jat asam panas dikipas dari sana. Beri daku sepotong daging bakar, lenguh kerbau dan sapi malam hari. Beri daku sepucuk gitar, bossa nova dan 3 ekor kuda. Beri daku cuaca tropika, kering tanpa hujan ratusan hari. Beri daku ranah tanpa pagar, luas tak terkata, namanya Sumba.
Di Pulau Sumba, padang rumput luas terbentang karena iklim kering sepanjang tahun. Rerumputan nampak hijau namun berwarna coklat keemasan ketika kemarau datang. Itu pun di daerah pantai dan daerah aliran sungai, sungguh ideal untuk habitat ternak seperti kuda, sapi, dan kerbau. Puluhan ternak bebas berkeliaran di padang rumput yang luas. Sungguh pemandangannya luar biasa. Pantai yang mengelilingi Pulau Sumba juga tak kalah indah. Laut yang membiru dengan perubahan warna langit saat terbit dan senja membuat pemandangannya semakin eksotis. Sebuah puisi karya Taufik Ismail menggambarkan keindahan Pulau Sumba begitu berkesan. Lembaga Ilmu Pengetahuan Indonesia melakukan ekspedisi Bioresources Pulau Sumba untuk mengungkap keanekaragaman hayati di Pulau Sumba. Sebuah pulau yang dinyatakan sebagai daerah tertinggal. Tetapi apakah tertinggal juga keanekaragaman hayatinya? Berbagai versi sejarah geologi Sumba menjadi misteri alam yang tersembunyi oleh berlalunya waktu. Menurut sejarah geologi, Sumba dahulunya merupakan sebuah fragmen lempeng benua yang bersambungan dengan dangkalan Sunda. Teori lain mengatakan Sumba dahulunya pernah bergabung dengan Sulawesi Barat. Ada pula yang mengatakan Sumba merupakan pulau oseanik yang tidak pernah bergabung dengan pulau lainnya. Ekspedisi ini akan membantu menguak masa lalu dan memberikan pemahaman bagaimana evolusi yang terjadi di Pulau Sumba ini.
Tahun 2016 kita melakukan eksplorasi bioresource itu di Pulau Sumba ini. Ini dikarenakan karena Pulau Sumba itu memiliki keunikan yang hampir tidak kita temui di kawasan lainnya di Indonesia. Menurut teori pembentukan Pulau Sumba sendiri, itu ada beberapa teori yang berkembang, yaitu bahwa Pulau Sumba ini dahulunya berada di bawah permukaan laut, dan itu sebelum dia terpisah menjadi pulau sendiri, itu bersatu dengan Sulawesi bagian barat, kemudian ada sedikit bagian di Kalimantan Selatan. Nah, itu kemudian beberapa puluh ribu tahun yang lalu itu baru terpisah gitu. Nah, di sini kita berharap bisa membuktikan argumen kita tersebut melalui organisme-organisme yang kita temui di Pulau Sumba ini.
Penelitian dilakukan di hutan dan kawasan pedesaan di area Taman Nasional Manupeu Tanah Daru dan Laiwangi Wanggameti, Kabupaten Sumba Timur, Nusa Tenggara Timur. Dengan topografi berbukit-bukit, dengan sungai berada di lembahnya yang curam, Sumba Timur mampu memikat siapapun yang berkunjung. Di bagian tenggara, terdapat Gunung Wanggameti, yang merupakan gunung tertinggi di Sumba, yang juga menjadi salah satu lokasi penelitian. Pulau Sumba merupakan bagian dari wilayah Wallacea, wilayah yang memiliki fauna dan flora unik percampuran flora fauna Asia dan Australia. Saya Australia. Sayangnya, keanekaragaman hayati pulau ini pun belum banyak diketahui, meskipun eksplorasi ilmiah telah dilakukan setidaknya semenjak tahun 1910. Hamparan luas padang rumput di Taman Nasional Manupeu Tanah Daru dan Laiwangi Wangameti membentang luas dari satu bukit ke bukit lainnya. Belukar atau pepohonan kecil hingga sedang dapat ditemui antar bukit atau ceruk, di mana dapat juga ditemukan sungai atau aliran air. Di kawasan Wangameti, ekosistem hutan pegunungan dan padang rumput yang luas dan berbukit menjadi target utama penelitian, sedangkan vegetasi basah yang berada di hutan dataran rendah, hutan tepian aliran air, hutan pantai, serta bukit kars menjadi target utama penelitian di kawasan Laiwangi. Salah satu keanekaragaman tumbuhan tinggi endemik yang dimiliki Sumba adalah sikas, yang merupakan grup tumbuhan berbiji terbuka yang tergolong sebagai kelompok tumbuhan awal. Sikas sundaika yang ditemukan di Sumba Timur ini merupakan jenis endemik di Kepulauan Sunda Kecil. Lulu Ura,
Selamat pagi
masyarakat
Bu Titi, sudah hadir ya?
Bambu ini di Sumba Timur. Tumbuhan yang menarik karena bambu ini tidak tegak seperti bambu umumnya, namun merambat hingga mencapai ketinggian 30 meter.
Selamat pagi. Sudah ada.
Selamat pagi Bu Titi, apa mau mencoba share screen atau kita yang share screen atau mau Ibu yang share screen?
Saya sendiri aja yang share screen ya.
Oh boleh?
Ya. Cukup jelas kan suaranya?
Suaranya sangat jelas Ibu.
Ya.
Kameranya juga clear kok.
Ya.
Ibu Eis juga
Boleh.
sudah ada di sini.
Iya.
Yang akan memandu nanti sebagai moderator.
Baik.
I'm really sorry, I forget to speak in Bahasa. Good morning, Ibu Titi. Once again, welcome to Isibio. So, are you ready to start your session? So, please be reminded that some participant is already joined us, so they will see your presentation.
Udah tunggu aja tuh.
Oh ya, oke. Ya, boleh.
Good morning, Ibu Eis. Are you ready?
Yes, I am. Thank you.
Good morning, ladies and gentlemen. The symposium is going to start in 5 minutes. Please make sure that you have a stable connection. I'm Eva Agustriana, and I will accompany you through the session. Before we begin the program, I would like to tell you about the conference rules. Here are the conference rules. Oral and non-oral participants are expected to join at least 30 minutes before the conference is started. All participants are expected to join as much as possible all sessions in this conference. Please use the virtual background provided by the committee. Please rename your Zoom username according to the following format. For oral participants, you have to assign your username as code of room R1 or R2 underscore code of presenter underscore name. Code of presenter can be checked in the oral schedule program at the column code. For non-oral participants, you have to assign your username as code of room underscore participant underscore name. Please mute the microphone, especially during the presentation of keynote speakers. In the parallel session, oral participants should activate the camera and microphone during their presentations. Questions from all participants are delivered using the chat feature in Zoom according to this format: name/institution/ the name of the speaker /question. Every session will be led by a moderator. Moderator will decide which question is to be discussed or answered. All participants have to fill the list of attendees provided by the link given in each session. Attendees are not allowed to write information that is offensive, racist, or immoral in nature. Organizing committee has the full right to block or remove any attendee that break the above rules. Only participant who followed all the sessions in this conference is given the certificate. Ladies and gentlemen, we will initiate the program. Assalamualaikum warahmatullahi wabarakatuh. It is a great pleasure to welcome you to the 7th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2020. November 18 to 20, 2020. The 7th Isibio is highlighting the sustainable use of plant, animal, and microbiome for advancing human welfare and natural conservation. The event is hosted by Indonesian Institute of Science, LIPI, conducted virtually and live from Zoom meeting room, an official YouTube channel of LIPI. It is a great honor for us to introduce and welcome the honorable guests. Dr. Laksana Trihandoko, Chairman of the Indonesian Institute of Science, LIPI. Dr. Yan Rianto, Acting Deputy of Life Science, LIPI. Professor Dr. Oki Karna Rajasa, Deputy for Earth Science, LIPI. Dr. Puspita Lisdianti, Director of Research Center for Biotechnology, LIPI. Dr. Atit Kanti, Director of Research Center for Biology, LIPI. Dr. Arhadrian, Director of Research Center for Plant Conservation and Botanical Garden, LIPI. Dr. Iman Hidayat, Director of Research Center for Biomaterial, LIPI. Dr. Ratih Pangestuti, Head of Marine Bioindustry, LIPI. And all honorable speakers and participants. First of all, let's say thanks to God who has been giving us guidance, happiness, healthy, and mercy, so we can attend and participate in this event without any obstacles. Ladies and gentlemen, the first agenda, National Anthem, Indonesia Raya. Ladies and gentlemen, we will start the International Symposium with warm welcome remarks from the Acting Deputy of Life Science, LIPI, Dr. Yan Rianto. Dr. Yan Rianto, the time is yours.
It is my pleasure to welcome all of you in this very important meeting, the 7th International Symposium of Innovative Bioproduction, Indonesia on Biotechnology and Bioengineering 2020. Since then, the meeting was first held in 2013 and has been annually carried out by Research Center for Biotechnology. This year, Isibio entering a new stage with some change for better elaboration. For the first time, the meeting is held virtually. The second, this meeting is organized by collaboration between Research Center for Biotechnology, Research Center for Biology, Research Center for Biomaterial, Research Center for Plant Conservation and Botanic Garden, and Research Center and Research and Development Division for Marine Bioindustry, Indonesian Institute of Sciences. Lastly, the meeting terms are broader to cover biodiversity and the use of bio-based material and biotechnology and bioengineering to produce bioproducts. For this reason, the theme for this symposium is sustainable use of plants, animal, and microbial for advancing human welfare and nature conservation. Distinguished ladies and gentlemen, Indonesia has long been known to have very high diversity. We are known as mega biodiversity country. As we all know, biodiversity is essential for human health, economic, food safety and security, energy alternative, and environmental health for individual or broader aspect of human welfare and well-being. Conservation of biodiversity is as important as its sustainable use. Our mutual challenge are to take full advantage of scientific and technological development to explore the potential of biodiversity. This symposium will elaborate 5 topics, including biodiversity and conservation of natural resources, biodiversity utilization for human health and welfare, bioprospection of natural resources, genomic application to promote the use of natural resources, and biotechnology and bioengineering for bioproducts. We have received 94 full papers from various research institutions and universities. To cover all of the topics, all of the paper will be distributed in every topics. Distinguished guests, participants, ladies and gentlemen, in this excellent opportunity, I would like to show my greatest appreciation for all of our keynote speakers from Indonesia, Singapore, Brunei Darussalam, Japan, and United Kingdom, who are willing to share their experience and excellent research. I would also like to welcome all presenters and participants to deliver, share, and update their research result. To end my remarks, I welcome you all to participate in this symposium actively. I hope that we can exchange our knowledge, experiences, and research results to ensure the sustainable use of our biodiversity for the future generation. Thank you very much.
Thank you very much, Dr. Yan Rianto, for the opening remark. We would also invite Dr. Laksana Trihandoko to deliver an opening remark. To Dr. Laksana Trihandoko, the time is yours.
Hello, I am Handoko, the chairman of the Indonesian Institute of Sciences, LIPI. It is a great honor for all of us at LIPI to host and to organize this international conference. On behalf of LIPI, I would like to welcome you all at this international conference. We hope this international conference would open an opportunity for all of you not only to know each other, but also to open future collaborations. I would like to thank to the distinguished invited speakers and all participants for your contribution. Have a nice, fruitful, and productive discussion during this international conference. Stay healthy. Thank you very much.
Thank you very much, Dr. Laksana Trihandoko. Before continuing to the session, I would like to inform you that for the next session, we will have 2 lectures delivered by Dr. Slik from University Brunei Darussalam and Dr. Sunarti from IPB University. The session will be hosted by Professor Ais Hermiati. Professor Ais Hermiati is a researcher from Research Center for Biomateriality with the expertise in the field of biorefinery, biomass, and lignocellulose. Professor Ais was graduated from IPB University, majoring food industry technology for her doctoral degree. To Professor Ais Hermiati, the time is yours.
Okay. Thank you very much, Ms. Eva. Good morning, ladies and gentlemen, all participants of Isibio 2020. We have a little bit early because in the schedule it should be started, the plenary session should be started at 9:40, but now it's still 9:10, but it's okay. I think we can just start the plenary session. It is my honor and my pleasure to be a moderator for this plenary session. In this session, we will have 2 keynote speakers, Professor Johan Willem Frederik Slik from the Faculty of Science, University Brunei Darussalam, and Dr. Titi Chandra Sunarti from Faculty of Agriculture Technology, IPB University. Professor Slik, or Ferry Slik, will deliver a topic on biodiversity conservation in Southeast Asia, while Dr. Sunarti will deliver a topic about the application of biotechnology and bioengineering for carbohydrate bioconversions. Both topics are such important and interesting topics in this space. In this plenary session, each speaker will have about 40 minutes for presentations, and at the end of the two presentations, we will have about 20 minutes for discussion. Participants may type his or her questions or comments in chat. Please mention your name, institution, and to whom the question or comments are addressed to. Then you may type the question or your comments. Participants may also raise hands during the discussion sessions. Before we listen to Professor Slick's presentation this morning, let me read the CV of Professor Ferry Slick. Professor Slick got his master's degree from Leiden University in 1994 and got his PhD degree from the same university in June 2001. His research experience after graduating started as a researcher at National Herbarium Netherlands, Leiden University in the Netherlands from 2006 until 2008. From 2008 until 2013, he is a professor at Chinese Academy of Sciences. Starting in December 2013 until now, he is an associate professor and curator at the University Brunei Darussalam Herbarium, University Brunei Darussalam, Brunei. Professor Slick is a member of the Association for Tropical Biology and Conservation and committee member of the Brunei Nature Society. He involves in some research projects related to biodiversity. He is also an editor in some scientific journals and a curator at University Brunei Darussalam's Herbarium and Herbal Garden. Professor Ferry Slick has also published many papers in scientific journals. Now, I would like to invite Professor Ferry Slick to deliver his presentation. Professor Slick, you have 40 minutes for your presentations. Please, the time is yours.
Okay, is it visible? And am I clearly, can you hear me clearly?
Yes.
Okay, thank you. Good morning to all the participants. And well, I feel honored to be invited as a keynote speaker here. What I want to talk about is about biodiversity conservation in Southeast Asia. And we're actually at a very interesting time, but also an alarming time, because, well, we all know the biodiversity, we are in the midst of a biodiversity crisis. But the nice thing for us biologists, at least, is, and also for interdisciplinary research at the moment, is that it's also a time where we are, well, better than ever, capable of monitoring the changes that are happening in the environment. So this talk is going to be first maybe a bit depressing because it is about the crisis, and then hopefully afterwards a bit more positive because it's going to be about all the new developments that we have available at the moment. Okay. So here I have a map of the, well, I focus mostly on plants because plants are my favorite organisms. And so this is a map of the world, where you can see the biodiversity hotspots for plants. And you can see, of course, the tropics are very diverse, like here in South America. There's a bit quite high diversity in the Mediterranean region, in tropical Africa, also in South Africa and Madagascar. Then this more or less tropical, subtropical transition in East Asia, and especially important for us here during this meeting, this very high diversity spot, which mostly comprises Indonesia. So Indonesia is a very important country for biodiversity globally. Now, I think we all know it's not going that well with the biodiversity at the moment. This was actually, these are a few titles of papers by biologists from the early 2000s, where they are already warning that things are not going that well. So here are some quite alarming titles like Southeast Asia, biodiversity and impending disaster, tropical turmoil, biodiversity tragedy in progress, and momentum drives the crash, mass extinction in the tropics. You can actually also see that in these kind of maps. I think many of you may have seen these maps. I still like to use them, although they are a little bit outdated. But they show very clearly what kind of problem we are facing. So, for example, if we look at Southeast Asia, mainland area, for example, in 1970, and I always like to give the example of, for example, an elephant or a tiger. If you were an elephant or a tiger and you were living here, so almost in China, you could still walk through the forest all the way to Malaysia. Now, I don't say that that happened, but it was possible because most of the forest was still connected. So it also meant that the genetic exchange was still more or less continuous. Even though the animals may not have moved all the way from the north to the south, their genes might still have moved freely through the whole region. So you can see by 1990 that is as good as impossible. All the forest has been fragmented and all the forests are like little isolated patches. So they have become independent of each other. And the same thing is happening here in Sumatra and also in Borneo, where most of the lowland forest has disappeared over time. And at the moment, New Guinea is also a hotspot of change. Although this 2020 map was a projection, and I think actually currently it's not as bad as shown in this picture. Anyway, but this picture clearly shows what the problem is that we are facing. Now, sorry for my video, which is doing a bit strange. The main driver of this crisis is just simply human population density and growth. So people are everywhere at the moment. You can look at this graph here at the bottom. You can see human population densities were quite stable until more or less the introduction of antibiotics and modern medicine. And then human population started exploding. And you can see that most of the population increase up till present has happened in Asia. And you can also see that in this map, which actually means that the tropics in Asia are also among the most threatened in the world. So, because, well, all these people, they need a good living and they will exploit their environment to get this good living, right? So more people means more problems for the environment. Now we have many things that result from the high human population densities. This is, for example, habitat loss, fragmentation, and degradation. I have an example here from a district in China where I used to work. You can see that in 1988, most of the area was still green, and green means forest. And then we have these patches here of pink, which are rubber plantations at that time. Now you can see by 2003 and 2006, the rubber plantations have exploded and covered almost the whole area. And all the forest that was originally there is now fragmented. And actually, the big patches of green that are still there are only there because they were nature reserves. I'm quite sure that if they were not nature reserves, they would have been converted to oil palm plantations, rubber plantations as well. So when you drive along the road now, so in the past it was nice forest, but if you drive on the road now, you still see forest, but it's only one species, just the rubber plantation. So that has a big impact on biodiversity. You replace a very high diversity tropical forest with a one species monoculture of actually even an exotic tree species. So this is a big problem that is happening all over the region. Then we have this problem with fires, which are now almost happening at a frequency of a yearly frequency. This is an example of East Kalimantan in Indonesia, where we had these big fires in 1982-83, and again in 1997 and 98. This area, indicated in yellow, orange, and red, is the burnt forests or burnt area, which was more than, I think, 3.5 million hectares was burnt. But the interesting thing is when we look at all these fires, where did these fires happen? So we can see that a lot of fires actually happened in areas where the forest had already gone. So these were like these shrubby grasslands. The other fires happened in locked forests. So these were forests that were already disturbed by people, and then very few fires actually happened in the undisturbed forest. So this already shows what we knew, that if you have a closed primary forest, the chance of a fire is actually very low. But as soon as you start opening up the canopy of a forest, for example, by logging, the forest becomes much more susceptible to droughts, and fires can start more easily. So the fires are not only a result of these droughts, but also of the combined impact of human activities and the drought. Then on top of that, we have over-exploitation of our resources, wildlife and plants. And this is also related to the fragmentation of the forest, because the more the forest is fragmented, the easier it is to actually enter these forests and take animals and plants out of these forests. And Southeast Asia is actually one of the main illegal wildlife areas of the world. So trading areas in the world. So something really has to be done here, because the volumes that are traded are really huge and not sustainable. Another problem that we now have is invasive species. Invasive species often like areas that have already been disturbed. I have a few examples here. This is an example from Java, where a American secondary forest tree was introduced into the Bogor Botanical Gardens. Probably, we're not completely sure how it spread, but that's a likely source. And you can also see it spread from Bogor all the way almost already to Jakarta. So this whole area, you can now find this exotic tree. Now, this Cecropia is actually not such a big problem. It just mixes with the local species. It doesn't really cause any problems. So in this case, we are still lucky. But there are also examples like this, the water lettuce, which is an invasive also from the Americas, that can cover a whole water body. So this area, what I show here, is not a field of plants. This is a lake completely covered by the water lettuce. Now you can imagine what happens if this plant is covering the whole lake. There will be no more light going to the bottom of the lake. So the whole ecosystem of that lake is completely changed because of this plant. And because it grows so fast, it's also very difficult to get rid of it. So invasive plants, also a problem. Then, of course, we have pollution itself, like pollution coming from mining, here also gold mining, for example, or people just putting their rubbish in the rivers, and not only people, but also industries, or pollution like here, like from infrastructure and heavy industries. Now, another problem is land use planning and law enforcement, it is actually, in principle, very well possible to develop an area for a specific crop like oil palm or rubber plantation, but still leave forest in between the plantations, so that, and especially if you can do it in a network way, and so that the biodiversity can be maintained within this productive landscape. But what often happens in the tropics is that the development of these plantations, even though perhaps they may have been planned well, the execution often results in complete change of the landscape, with the whole landscape covered in this plantation area without any forest remaining. Here I have an example from Indonesia. It's Gunung Palung National Park, which is a very national park because it contains a mountain and it runs all the way to the coast. So it actually contains most of the forest types that you can encounter in Borneo in a single national park. You can see what happened in 1988. So this is the park, the yellow line. The park was still more or less intact, although there was already some human activity here on the east side of the park. So these red parts are areas where the forest has been cleared for mostly oil palm, I think. And this is the situation in 2002. So lots of forest has disappeared around the park, but you can see also inside the park. So this is not supposed to happen. So this is a problem with law enforcement. So this is an example from Indonesia, but these things are happening all over Southeast Asia. So this is also a major problem. And then on top of that, of course, we have climate change. So the whole setting of all the forests and plantations will change in the future because the temperature will go up and the rainfall patterns may change. We may get more intense droughts or floodings. So that will also greatly affect what will happen in the future to the environment. So this is a map from Indonesia, and it shows which areas, well, Indonesia and surrounding region, which areas are especially susceptible to climate change. And Java actually scores quite high. Okay. So this was the depressing part of the talk. Let's switch to the exciting part. So while the environment is degrading around us, our possibilities to study this and monitor this over time have increased dramatically. So for biologists, this is a very interesting time because we have now so many possibilities to study the environment. So one of the things that have happened over time is, well, of course, the internet. And well, this whole meeting is an example of that, right? We're talking over internet. So more and more species data have become available online. And I can remember when I was a PhD student, which is not so long ago, so that was around the year 2000, I really struggled to find information of the tree species that I had collected in my fieldwork in East Kalimantan. I had to go through books, ask experts. It took a lot of time. But nowadays you can just type in all these species names and you get information almost right away. So I have some examples here. Maybe you know the Global Biodiversity Information Facility, where you can find plant and animal collections around the world. So it's very easy to see where the species is distributed. There are these very large databases developed now, like the TRI, the plant trade database, where you can just download information that you need for your study. There are now websites where you can see pictures and descriptions of plants, etc. So also several examples from Indonesia. So this is already a great help. A lot of this data is now also used for applications like this, automatic identification tools. So where you can use your mobile phone to take a picture of a plant and the plant will be identified for you, or they will find something that looks similar to it, so it may help you identify a plant. Now, many of these applications work quite well in temperate regions where there are not so many species, but there is still a big challenge for biodiverse, rich regions like Indonesia, for example. We are trying to make something like this for Brunei, but even in Brunei, we already have 5,000 species of plants. So it's a huge task, but it's really a nice challenge to try because it will involve people more with nature, right? If you can just use your phone to take a picture of a plant or animal, and the phone will give you the information about the species and what it does. Okay, this is also really exciting what is happening. This is actually already quite old, DNA barcoding. DNA barcoding is the sequencing of a very small part of the DNA that is very easy to sequence and very cheap because it's so short region. And they have done this for animals first. Plants is slightly more difficult because for plants you need 3 different regions to get a good identification, but for animals you only need 1. And this really is a big help for several reasons. Well, one, for example, I have an example here. This is one of the first studies where they used DNA barcoding. So this is, I think, already more than 20 years ago, was to find out which caterpillars fitted with which butterflies. That's actually a big problem because the only way to find out, especially in the tropics where there are so many species of butterflies, to find out which caterpillar belongs to which butterfly in the past was to breed the caterpillar and wait what kind of butterfly would come out. But now we can just sample the caterpillars and the butterflies and directly link them together. And well, there are many examples like this. One of the most exciting ones, in my opinion, is that using this technology, we can actually also identify microbes that live in the soil. So suddenly we can identify the bacteria and the fungi living in the soil. So this is something that I would like to do here in Brunei. And I think in Indonesia, there are also many possibilities for this. So you can just take little samples of soil and sequence them and find out what species, so how many species of bacteria and fungi there are, and also what species there are. And you can compare between different areas. You can actually also study the roots of trees using this method, because normally when you dig in the soil, you can find the roots, but it's almost impossible to find out what species of plant the roots belong to. But using the barcoding, you can easily find out. So really exciting. Even more exciting now is the genomics, so the next gen sequencing, where we can actually just sequence the whole genome. And these technologies have become so good that it's now even possible to collect a sample of water from a river and still find in that sample the DNA, so that's called environmental DNA, of species that live in that river. So you can do an inventory by just collecting some water. So this is also an exciting new development. But if I come back to that soil microbes example, one thing that we can do with these new technologies is not only identify how many species of microbes there are and what species of microbes there are in the soil, but we can also extract direct information on the genes that are active within the soil. So we can get a functional picture of what is happening in the soil. Because with these technologies, we can recognize genes that are present and active in the soil. Okay, so really exciting possibilities here. One of the other nice things that we can do once we have these DNA sequences is e-taxonomy, which makes the work of ecologists and also taxonomists much easier. So in this example, I have a plant specimen from an herbarium, but it could also be a living plant from a plot. anything is possible that has DNA. So once you have the sample, you extract the DNA, or you have many examples from a plot, for example. You extract the DNA, you sequence it, you can compare how similar they are, for example, using BLAST. Based on the similarity, you can group them into sequence clusters. So these sequence clusters are species that have very similar DNA. So they are most likely very closely related species within the same genus or family. So once you have that, you can take a look at your actual specimens again and see how well the computer has grouped your specimens. So this can be done very quickly, because normally when you are identifying plants, or even worse, if you are identifying insects, for example, it can take weeks, months, sometimes even years to identify all your material. Well, this kind of technology can do it within, well, several hours to a week, perhaps. So it saves a lot of time. Even if you don't know the name of the species, but you just have these sequence groups, you can still do lots of ecological analysis. You can compare the similarity in sequences between different sites, etc., or the diversity of sequences in different sites. And another thing that you can do is, because this is DNA sequences, you can also use that to produce, for example, a phylogeny of the species in your sample, which is also interesting for biodiversity research. So that's this example, for example. So here we have a tree species diversity map of Borneo that I made quite long ago already. So it may be outdated already, but still you can see that this mountainous region in Borneo has quite low diversity of tree species, while the lowland areas, which are actually also the most threatened areas, have quite high tree diversity. So this is the traditional diversity map that we could make in the past. Now, once we have the phylogeny of all these species, well, in this case it was genera in these areas, and we combine that with the plot information, we can actually turn the phylogenetic information into a phylogenetic diversity map. And then we get this map of Borneo, where actually these mountainous regions show up as the areas with the highest phylogenetic diversity, which means that in these areas you find groups of species growing together that are either very old lineages or they are very distantly related. While in the lowland areas, where you find these whiter colors, so lower phylogenetic diversity, you often find species growing together that are closely related to each other. For example, many species of dipterocarps. So this suddenly gives two different views of the diversity of Borneo, which are useful for conservation. Now, remote sensing is another very exciting field that we have nowadays. We can do really nice things now, and this one is one of my favorites, because once we can do this for the tropics, so this is also very nice for Indonesian to try. We can suddenly do tree inventories for whole very large landscapes, or for example, the whole of Indonesia if needed. So this is based on specific frequencies of light that plants reflect. And it turns out that almost every species of plants, so here we see a list of different species of plants, has their own specific reflectance. So you can actually identify specific species of plants just from a picture that you take with the right frequencies, of course, of the forest. So here there's an example where they have identified the trees in the picture using this technology. So this has some very nice applicabilities, because, well, not only can we map species compositions over very large areas and calculate the diversity for every area using this method, we can also pick out specific species, like here. For example, if we have a problem with an invasive species somewhere, the old way was to first you had to find all these invasive species in the area, and then you could eradicate them. But using this technology, just taking one picture, you can immediately identify where all these plants are. So it makes it much more efficient to eradicate that species, if that is the aim. So really exciting things. I hope I can also do this in Brunei. This is already a bit older, but still being more perfected. This is also remote sensing using radar. So with radar, you can actually measure the height of the ground level and the height of the canopy. And the height of the canopy is often directly linked to the amount of biomass in the forest. So by just taking a radar image here for this mountain in Hawaii, you can immediately calculate the biomass pattern for this forest. So these red areas have high biomass and the blue areas have low biomass. So if you can do this for a whole country, you can actually very exactly calculate the amount of carbon stored in the forests of these countries. And nowadays, these you also have handheld or ground-based radar technology, so you can actually take very nice three-dimensional pictures of trees using this technology. Okay, most of these remote sensing equipment used to be quite large, so could only use it on planes or on satellites, which made it a bit complicated. But nowadays they have become much smaller and you can actually fit them on drones. So drones, conservation drones in this case, they are also a very important new development because it makes it possible for simple researchers like us to use this fancy technology to study the forest. This is a very early example from Sumatra where they were looking for orangutan nests and they were still using these planes instead of the, how do you call these, quadro helicopters or something that people use nowadays. But well, the nice thing is you can just program the course for this drone and it will fly this course by itself and then come back with all the camera images that you want to get. Okay, a very important development over the last few decades is also the availability of long-term plot data. So often people make plots in forests, and if these plots are permanent plots and you keep monitoring them over a very long time, suddenly you become able to see how the forest is changing over time. So this is different from establishing plots just once and then never visit them again. This is really monitoring plots over a very long time. So some of these plots have now been monitored for several decades, and we are finding really interesting patterns. So in the past, people often thought that tropical forests were quite stable environments, but it turns out that they are very dynamic. So one of the things that has been happening in the Amazon region, for example, but probably also in Africa and Asia, is that the lianas have become more common because it's probably related to the climate change, because we can also see that the tropical forests in South America and also in Africa, and slightly less so in Asia, have been increasing in biomass. So the forests have actually been taking up a lot of carbon dioxide from the atmosphere and turned it into wood. So you can see here from the 1980s to early 2000s, the amount of carbon stored in tropical forests just increased and increased and increased. This is probably because the carbon dioxide concentrations in the atmosphere also increased, and carbon dioxide is a very important resource for photosynthesis. So in the past, probably carbon dioxide had been limiting for tree growth, and because we are adding carbon dioxide, it's actually good for tree growth. But you can also see that from the early 2000s onwards, although this graph doesn't really continue much into the present, this process has stopped and in some cases even reversed, because when the temperatures become too high, this is actually bad for photosynthesis. So trees start taking up less and less of the carbon dioxide. Okay, so all these things are possible because of these long-term monitoring plots. So I really encourage the development of a very nice plot network across Indonesia. Would be really informative. Other things that you can do with plot networks are here. This is at a global scale. These are hundreds of plots, and we just mapped the tree diversity. So you can recognize a very diverse area here in the Amazon, some diversity here in Africa, but also that, for example, that this area in Indonesia with Borneo, Sumatra, and the Malay Peninsula is as diverse as this very diverse region here in South America. So really exceptional diversity. And you can also compare using these plots how these forests differ from each other. So we can really see here that the South American forests are really different in species composition than the Asian ones. And we can even see the gradients of difference between the different forest types. So Africa is somewhere in between. Because we are also collecting lots of data from, for example, the oil industry, they are drilling, so they take soil samples, which we can use for pollinology. So we can reconstruct vegetation types in the past. And because our ability to reconstruct past climates also has increased dramatically, we can actually see how the different regions, so this is an example of Southeast Asia, where you see Borneo here, Sumatra here, and Peninsula Malaysia here, how these regions look in the past. So this is during ice age, so the land area looks completely different because Borneo is connected to Southeast Asia and Java and Sumatra and Malaysia because the sea levels were down by about 100 meters. But because we can reconstruct the climate and we also have the pollen data, we can actually see what kind of forest there was where in the region. We can nowadays also do the same thing for the future. So this is a map of Europe, where under a certain scenario of climate change, they have predicted how the species composition will change into the next few decades. And then you can identify regions like these red regions where the species diversity will go down, and other regions like these grayish regions where the species diversity will increase. And they can also predict what kind of species will decrease and what kind of species will increase. So people can already anticipate these changes that will happen in the future under climate change. So that is very important for policy development. Very important as well are secondary. Okay.
And in our research, we have found that actinomycetes also can eliminate the lignin in the biological pretreatment. And of course, some microorganisms produce enzyme such like ligninase or hemicellulase and cellulase in the from the biological treatment. Some chemicals also will remove the lignin and also the lignin, the hemicellulose from the structure of the biomass in using the alkali, the acid, explosion, gas, oxidizing agent is also with the solvent extraction. So we can choose which one is the pre-treatment is proper with our biomass. It's really depend on the characteristic of the biomass. In detail, we will see the some pretreatment to improve the enzyme susceptibility, especially for the cellulose and also for the starch. Cellulose and starch they have almost similar structure because both of them are the polymer of the glucose, but they have different in the linkage between each monomer. Such like in cellulose, they have beta 1,4, but in the starch, they mainly having the alpha 1,4, and also the branching linkage is with the alpha 1,6. So we can see some research about this, and let me see. Let me show you. Okay. In this research, we found that the milling process can reduce the particle size of the sweet sorghum bagasse. In this research, we can see sweet sorghum bagasse was milling and separated into 3 fractions, less than 250 micrometers, and the bigger particle more than 420 micrometers. And this is the between the higher and the smaller one. And the cell wall was broken. Maybe this is the effect of the pressing during the juice extraction, and also during the milling process. Sometimes a hammer mill or disc mill give a lot of energy and makes the friction between the particle also will produce energy, heat, and it can also broken the structure of the fiber. And from this table, from this graph, we can see that the smaller particle have more porous, as we can see this one. The higher one is a little bit smooth, yeah, but we can see there are some destruction is made the smaller particle have the more porous and high surface area, so it becomes easy for them to the enzyme susceptibility. The graph shows that the smaller particle with the excess enzyme, this is the cellulose enzyme, can produce higher polysaccharide conversion and also higher reducing sugar yield if we compare to the other particle size. The pretreatment by using the alkaline solution and also hydrothermal. Hydrothermal, it means high pressure and high temperature, also will increase the purity of the cellulose. This picture shows us that the more higher crystallinity, it means it more high level the purity of the cellulose. And by using the alkali hydrothermal, the purity is higher compared to only the native one. This is the native bagasse. And if we only boiling in the hot water and compared to the boiling alkaline water and only hydrothermal without alkaline solution, so the alkaline hydrothermal give the best purity for the cellulose fraction. This microscopic observation also we can prove that the native, this is the native, is still have the natural structure, but only by using the hot water, maybe the extractive process, the extractive fraction of the extractive component removed, but in alkaline there are some destruction, maybe the lignin also removed, and by using the hydrothermal, a little bit higher temperature, it's a little high pressure, also broken the structure. But using the alkaline, we can see there are a lot of you can see this one, we can see a lot of porous produced after this treatment. So the effect of the alkali and hydrothermal also influence to the enzyme susceptibility, actually, this treatment was given by using the alkaline hydrothermal, by using the excess enzyme, also will produce the higher polysaccharide conversion compared to the other. But with the limited enzyme, maybe not all the structure was converted. So we can say that in the excess enzyme, actually the alkaline hydrothermal gives the better the lignification process. This is also similar with the Kumar et al. With his finding, the effect of the alkaline hydrothermal on the biomass porosity and also to the internal surface. This is caused by the high removal of the lignin on partial solubilization of the silane. So I think the process, this is a good combination between the alkaline and also the hydrothermal treatment. Yeah. Okay. This slide shows the effect of the microwave treatment as a physical treatment on the enzyme susceptibility of the sago pit. Yeah. Sago pit is the inside part of the sago trunk. This is the main raw material for the sago industry, and the sago trunk consists mainly not only fiber but also starch. Yeah. As we see in this microscopic structure for the native sago, we can see this is the fiber, and this one, the smallest one with the blue and yellow structure, is the starch. Yeah. In the conventional heating, the heat is transferred to the surface of the material and into also the interior of the material by thermal conduction. But microwave, they are not formed of the heat. So the heat is comes from the interaction inside the materials itself. So the heating, it comes from the inside to the outside, but the conventional heating from the outside to the inside of the materials. In the microscopic picture, we can see that the native starch with the high crystallinity in fiber and also the starch. And then after it was gelatinized in the water and by using the microwave treatment, the starch was gelatinized. But not only the starch, the fiber also swelling, and they change the crystallinity into the more amorphous fraction. So the microwave treatment not only working to the starch, but also to the fiber to make it more susceptible to the enzyme susceptibility. If we compare to the dilute acid solution in the microwave treatment, maybe because the microwave treatment, sometimes with 100% power level, they can reach until 200 degrees Celsius. So there are some further process, especially for the glucose, they can convert it into the furfural or hydroxymethylfurfural. So that's why in this picture, we can see there are a black spot. This is identifying the browning component. Maybe it comes from the furfural or hydroxymethylfurfural. But in the dilute acid treatment, actually, the starch and also the fiber will more excessive heating, and it becomes more easier to the substrate to be hydrolyzed by using the enzyme. Okay. The microwave treatment not only gelatinize the starch, but also swelling the starch and the fiber. And the heat treatment, we compare between the autoclaving 121 degrees Celsius and microwave treatment, and then we combine the enzyme for the liquefaction because we combine the two enzymes with the almost similar temperature condition. In this research, we find the similar pH and also the temperature for both enzymes working. And then it was followed with the saccharification process. And then we combine also with the amyloglucosidase and also with the cellulose. And after the autoclaving, this is the autoclaving, actually the enzyme for the liquefaction is give more susceptible with the microwave treatments products because they can having more total soluble sugar compared to the autoclaving. But after the saccharification, both of them give almost similar, even some of them slightly increase in the microwave treatment, but they give still higher degree of polymerization of the polymer. So actually both of them having the similar process. But autoclaving takes a very long time. We need 15 minutes to conduct the autoclaving treatment, but using the microwave, we can a shorter time, and this result maybe we find only 30 seconds until 50 seconds is enough to improve or to gelatinize and swelling the starch and also the fiber. This picture shows the macroscopic structure of the sagopit residue after the enzymatic saccharification. We can see this is from the autoclaving. Autoclaving only working to the starch only, but the microwave treatment not only working to the starch but also to the fiber. So we can see the rigid structure of the cellulose was converted into more amorphous. So this is as an action of the microwave treatment. Besides the physical treatment, we can also combine the delignification by using several combination of the pretreatment method. We try to use the corn cobs. Corn cobs, half of the corn cobs carbohydrate is cellulose, and most of them is hemicellulose. So corn cobs actually, this is a good source for the hemicellulose, especially for silane. And now it becomes silane. It becomes an expensive raw material for making the xylitol for the pharmaceutical industry. But the rest or the residue from the separation of the hemicellulose, we can have the cellulose fraction. This is the corn cobs, yeah, with almost one ratio, the same ratio between the cellulose and hemicellulose. The lignified corn cobs, yeah, we remove part of the lignin and also the cellulose fraction, yeah. We eliminate the lignin and also the hemicellulose smudge, and the alpha cellulose, it was a modified, the cellulose fraction, we modified into the more amorphous fraction. These pictures also you can see, this is the native corn cobs with a natural fiber. This is the structure of the natural fiber, but after the lignification, the fiber it a little bit shrinkle because they lost the integrity by the binding of the lignin. Yeah. After we remove also the hemicellulose and another component, so we have the cellulose fraction with a straight fiber, such like in this picture. And then modification into the alpha cellulose also convert the straight fiber into more gel form. Yeah. The pure cellulose, and then we want to, it was inoculated by several cellulolytic bacteria. We can see the change of the fiber before and after the fermentation. before the fermentation, the cellulose was a straight fiber, and then after the hydrolysis, some crystalline structure of the cellulose was cut off and it becomes more amorphous. So we can see there are some smoke area or not a straight fiber anymore. There are some also we can see in this this picture. We can see a lot of holes, yeah, a lot of holes and pores was formed after the fermentation. And we can see that the enzyme produced from the bacterial cellulose also broken and also break the crystalline part into the more amorphous. And the graph, yeah, the graph from this picture, we can see that the cellulose fraction, yeah, cellulose fraction and also the lignified corn cobs, cellulose fraction is more susceptible
di PPI ya.
Corn cobs.
Internetnya lancar.
Now we will see some application of this method by technology and bioengineering in the process and the product development, especially for the carbohydrate commodity. Okay. First, about the corn flour production. Yeah. Corn flour, it was a traditional corn flour was produced from the corn grits. The corn grit was steeping for several hours, and during the steeping, actually there are some fermentations happens to the steeping process, and then after the fermentation, we draining the rest of the water, and we dry in the low temperature for 24 hours, milling into the corn flours. During the fermentation, this is without any introduction of the microorganism. By using the spontaneous fermentation, we found that a lot of the microorganism was growth, and for 48 hours. At the beginning, lactic acid we cannot find, but we can find only amylolytic bacteria. But after 48 hours, a lot of the lactic acid bacteria, also cellulolytic bacteria, amylolytic, and also the yeast. But in the next research, we want to eliminate the amylolytic because the amylolytic will reduce the yield of the corn flour production, and the yeast also we avoid this this microorganism because it will convert some part of the component into the ethanol or acid. So we use only lactic acid bacteria and also cellulolytic bacteria as indigenous microorganism as a starter in the next research. Okay. After we isolate the indigenous microorganism, it was identified as lactic acid bacteria and the cellulolytic bacteria. And then we try to compare with the spontaneous fermentation and also using the only lactic acid bacteria, cellulolytic bacteria, and then combine between both of the microorganism. And then we found that the fermentation actually can be reduced if we found that the spontaneous fermentation need 48 hours to reach the perfect steeping, but the introduction of the microorganism will reduce from 48 hours to 12 hours. And the result from this showed that the crude fiber was decreased and the yield also will increase. So this is one application of the microorganism to improve the process and the product process efficiency and also the quality of the products. In the traditional corn flour, sometimes they only having short stepping process. Yeah, sometimes only 3 hours to let the grits absorb the water from the stepping water. But this condition only can have a low yield in the percent of the floor yield. This is less than 60% of the yield of the floor. So we try not only by using the microorganism, but also by using the vegetable enzyme. In this research, they use the papain to improve the process efficiency, especially the enzyme, the cellulosic enzyme can reduce the grit hardness, and also it make it easy to milling the grits to make it as a floor. So when we use the excess enzyme until 1% of the stepping solution, they give almost 100% of the floor. It means almost all the grits can be reduce the size to make it the floor. And this graph also showed that the grit hardness reduced markedly with the addition of the enzyme from the papain. Yeah, if there's no enzyme addition, actually stepping only absorb the water, but also it will swelling the grit, but not as much as the treatment with the papain. In the sour cassava starch, the sour cassava starch actually is not a native Indonesian product. It was in South America, and in South America, it was famous as polvillo azedo. It comes from the spontaneous fermentation of the cassava starch, and it was fermented in a tank. It takes very long time, sometimes more than 1 month to the fermentation, and then it was naturally sun exposure by using the sun drying. It should be sun drying, and then they have the sour cassava starch. In Indonesia, sour cassava starch is useful, especially in the traditional snack or kerupuk, because in small scale industry, they prepare by themselves the cassava starch, and it was a very simple preparation, but it takes time. During the spontaneous fermentation of the cassava starch, we can see that the total acidity of the cassava starch is increased. Yeah, but and also it was followed with the lower pH. If we refer to the standard national Indonesia, maybe this sour cassava starch cannot fulfill the requirement for the SNI, but this is a typical cassava starch. They have a typical also functional properties. So it was different if we want to make it as a standard, because it has total acidity and also lower pH. During the fermentation, lactic acid bacteria also dominate the process, and also the yeast and the fungi, because naturally this microorganism actually we can find in the fermentation of some carbohydrate commodity. We try to introduce the indigenous lactic acid bacteria to the sour cassava starch production. So we isolate and then we culture it to make it more easy to the cassava starch to be converted as a sour cassava starch. Yeah. Compared to the native, to the spontaneous fermentation, actually the spontaneous fermentation takes around 30 until 50 days. So the introduction of the lactic acid bacteria will reduce the fermentation time until 15 days. But the industry is still reluctant to use because it takes time, the 15 days. They want only maybe 1 or 2 days, but it's very difficult for us to get the higher acidity and also the lower pH. Yeah. But the fermentation not only increase the expanding capability and also the swelling power. Swelling power, it means the capability for the starch to absorb the water, and also increase the solubility. Digestibility is lower, so it becomes a very good source for the resistant starch. The taste clarity also increase, so the introduction of the lactic acid bacteria not only shorten the fermentation time, but also improve the functional properties. Some traditional implementation of the cassava production is a small scale industry. So sometimes they give or use the river water as a water for the production. So a lot of the pathogenic microorganism from the supply water. So the introduction lactic acid bacteria also can improve the food safety because they can inhibit the pathogenic bacteria such like as Escherichia coli or Salmonella. Okay. This picture shows the microscopic from the light microscopic. This is a 0 days. There are no knot or porous in the center of the granule. But if we see the effect of the fermentation from the starch granule, a lot of spot or there are some hole inside the granule. It means that the lactic acid bacteria produce a lot of acid, and the acid also will make some erosion on the surface of the granule. And beside the lactic acid, some active compound also produced by lactic acid bacteria. It becomes a biopreservative for the products, so we can enhance the shelf life of the product. So it will a very good benefit for them to improve the quality of the cassava starch. Okay. This product shows that the increasing of the baking expansion capacity actually can be happen not only to the spontaneous fermentation, but also to the lactic acid bacteria fermentation. But we can see after 50 days, there are only slightly increasing in the baking expansion capability. But using the introduction of the lactic acid, the volume increased markedly. As we can see, this is in the picture. Maybe this is an almost famous product in Indonesia. They like to try such like a cireng, product of the cireng. So we try to measure the volume of the baking expansion, and for the 15 days, they have a very large expansion capability. Also, they have a more lower viscosity to make it more easy to be consumed or chewing the product. Yeah. It was a very interesting because some traditional industry, they try to make their own product, even a little bit sour smell, but it was have a good functional properties, so they still maintain the process. They cannot use the chemical preservatives such like antimicrobial, because if there's some chemicals, so there's no microorganism will grow in the spontaneous fermentation. So it make us can assume that the spontaneous fermentation, actually, it was also an organic process. In cacao bean fermentation, there are a lot of microorganisms take part in the formation of the flavor or aroma for the cacao. And in the cacao fermentation, this is from the spontaneous fermentation. The fermentation takes several days, and lactic acid will dominate the microorganism in the beginning or in the initial process. And the acetic acid maybe takes more than 3 days after the fermentation, but yeast also dominate in the initial process. A lot of product was produced during the fermentation. And there are some conversion from the fructose and glucose. The fructose and glucose, it comes from the pulp of the cacao bean. Yeah. So the fructose and glucose was converted into some intermediate products such like ethanol, acetic acid, lactic acid, also conversion of the fructose into the mannitol. All of the compound is important in the fermentation of the cacao bean, since it was contribute to the flavor formation. Okay, we can see some research. There are some. This is the some standard for the cacao bean quality. In the farmer, cacao bean fermentation conduct using the spontaneous fermentation, and this is fermentation produce low quality of the bean, and it was give lower price with low flavor and aroma of the cocoa product. So it was giving a very low added value to the farmer. So the fermentation, spontaneous fermentation, we try to compare with the lactic acid bacteria, the introduction to the lactic acid bacteria. Yeast, also the combination with between the lactic acid bacteria and the yeast. From the spontaneous fermentation, the fermented bean is very low. But if we introduce the lactic acid bacteria, the fermented bean was increased, and the combination gave the best result until 83% of the fermented bean has been produced from this fermentation. But unfermented also will reduce. Non-fermented, it means it's still raw cacao. Germinated, yeah, actually germinated cocoa bean, it was a poor quality because some of the component was converted into the new generation of the plant. So the germinated was they germinated was the part of the ethanol to inhibit the germination of the cacao bean. This is the slutty. It means give the bad flavor and aroma. We have to reduce as much as we can. So from the best fermentation, a lot of fermented we can produce, but we can also reduce the non-fermented germinated on the slutty coffee bean. As we can see, this is from the bean that we can produce from the fermentation. Yeah, this is a very good fermented, produce a dark, sometimes a purple color. Yeah, but it's just non-fermented. It's still a green one. So this is germinated. A lot of germs comes here, and also slutty. It becomes rotten one. The pH also become the alkaline. Actually, the cacao bean process, the alkaline process is a very important because they have a very good flavor and aroma if we want to use in the industry. Okay. And in the cafe, yeah, in the cafe, we also introduced some microorganism, actually from the Actinomycetes, as a starter for the coffee fruits fermentation, because we want to take some active compound from the coffee pulp. Actually, the coffee cherries, there are pulp and also the bean. And in the wet fermentation, they fermented the whole cherries. We did not separate the coffee pulp, but after the fermentation, we have to separate between the coffee pulp and also the coffee bean, so we can take the active compound from the coffee pulp to take some active compounds such like as a polyphenol, tannin, anthocyanin are also catechin. But we can see that the polyphenol was increased by the fermentation, but it can also reduce the bitter components such like tannin, increasing the anthocyanin, antioxidant activity, also the catechin as a part of the flavor in the coffee. So the introduction of the coffee, the microorganism, they can improve the quality of the coffee, green coffee, but also we can take some valuable product, especially the bioactive compound from the coffee pulp. Dietary fiber production from the cassava pulp also can be used. We can use some cellulosic microorganism. This is the cassava pulp. Cassava pulp actually from the small scale industry, they have two main component. It has a starch and also fiber. Yeah. So the fiber, we can make the fiber to make the dietary fiber. Cassava pulp in an inefficient process, they contain a lot of starch. So sometimes we have to eliminate, but we can take the starch component by using the enzyme. So we will have the glucose syrup. And then the residue is a cassava pulp fiber. We give the enzymatic treatment to produce a dietary fiber. Such like in this picture, the cassava pulp sometimes was dried, and it still can be used as the food component because it still have a lot of fiber and starch. Okay. This picture shows the scanning electron microscopic of the cassava pulp fiber before the hydrolysis. There are two treatments we can give, the thermal process and the non-thermal process. Non-thermal process, it means only boiling in the water, but thermal process, we use the autoclaving process. So this is from the cassava pulp fiber before the treatment, the enzymatic, from non-thermal and also to the thermal process. There are some destruction in the fiber we can find with the high thermal process, but after the hydrolysis by using the cellulose enzyme, a lot of the destruction will be exposed with the thermal treatment. So we can find there are a lot of degree of the value is reduced during the fermentation time. And also if we compare between the thermal treatment and the non-thermal, actually it's not so different. But if we find the functional properties, this is the soluble fiber and also insoluble fiber, they are not so different between the composition, but they can say that the functional properties, its water holding capacity, oil holding capacity, and also emulsifying capacity increase with the thermal treatment and the fermentation time. So the cellulolytic enzyme and also the thermal treatment will improve the dietary fiber properties and also the number of the soluble dietary fiber. As a conclusion, we can say that the biotechnology or bioengineering have important roles in the carbohydrate bioconversion, especially in improving the efficiency of the process, to improve the quality of the product, to produce valuable product, and to add the value of the agriculture materials, and to conduct the clean production, to eliminate the waste and the byproduct in agro-industry. Okay, I would like to, all the data presented in this lecture was produced from all my students' publication and also from my colleague. I would like to thank to all of them to support me in this presentation. Thank you very much. Terima kasih. Hello.
Thank you very much for Ibu Titi for the very brief presentations. We are really sorry because of the connection trouble. We're still waiting for Ibu Eis to join us again. Ibu Eis, are you ready?
Yeah, okay, ready. I'm sorry for the technical problem. Thank you very much for to Dr. Titi for the interesting presentations. Okay, for all participants, we have already listened to the presentation from Professor Slick and Dr. Titi. Professor Slick has already explained that we are in the situation of environmental crisis that affects biodiversity in Southeast Asia and other parts of the world also. However, on the other side, there are some new scientific developments that could help us to better understand, study, and monitor biodiversity and its changes. And Dr. Titi has explained about carbohydrate resources that are abundantly available and the role of biotechnology and bioengineering in the conversion and transformation of these carbohydrates into different kinds of useful and value-added products. Now I would like to invite questions or comments from the audience. I have seen one question from Dani Satyawan from ICA Biograd, is that right? To Professor Ferry Slick. His question is, what are the 3 DNA barcodes you mentioned that are needed to identify plant species with high certainty? Also, how good is the ability of nature to reclaim areas that we completely abandon? Is there any other questions? You may raise hands or okay, Dr. Slick, please just start answering.
I still have a problem with my camera. I'm sorry for that. The 3 regions for identifying plants, although it's not really my specialty, but I think it's RBCL, MAT-K, and what was the other one? ITS. And sometimes they had even a 4th one. So each of those, they can actually, they cover different levels of variation in the DNA. So the RBCL is very good in identifying family level, for example. And the MAT-K, or what was the other, TRNL, and especially the ITS, they are good for identifying genus, species, and sometimes even within species level differences. So it depends a bit on how detailed you want to identify your plants, but you have to keep in mind that there are several groups of plants, for example, orchids or gingers, that even if you use those 4 regions, often you cannot still not identify all the way to species. So it's like a tool that you can use for most species, but not always all species. So that's a bit problematic for plants. For animals, it actually works much better, and for fungi. And the other one for the restoration of areas, yeah, it's actually really good if you can follow the natural succession. Often, if the disturbance has not been too severe, or there is still natural forest nearby, you can often rely on natural regeneration. It's often even better than if you start interfering yourself, because when people start doing reforestation, they often don't get the species right, or the abundances of the different species within the new system are different from the original system. And also, when the regeneration is local, based on natural regeneration, you just get your original gene pool back as well. It's also important. However, if you want to help the natural regeneration, they have developed some good systems in Thailand, I think, where they have a set of standard species that they use, that especially species with fruits that can attract seed dispersers like birds or mammals. So they start out with those few species, and once they start flowering and fruiting, they will attract these natural seed dispersers, and other species will be brought into the system that you started with these basic, they call them framework species, I think. So if possible, I would always prefer natural regeneration. It's also much cheaper. But sometimes that's not possible. If there is no more natural forest nearby, you really have to start helping the regeneration. Okay, I hope that answered the question.
Okay, is that okay to Mr. Dani Satyawan? No? Yeah.
Very clear.
Pak Dani, is that okay?
Yeah, very clear. Thank you very much.
Okay, thank you, Pak Dani. Thank you, Professor Slick. There is another question from Dr. Ruliana Susanti from LIPI. What is the main challenges that you face when developing the plot network? In the case of Indonesia, we have so many permanent or semi-permanent plots in smaller size, less than 1 hectare. I was wondering how to connect all those plots when the methodology used is different. Please share your experiences. Please, I think you may directly answer the question.
That's one of the biggest problems that I always face when I do plot analysis. They all differ in shape and size, which can really influence the analysis, because if you compare like a square plot with a transect, you have a different level of beta diversity in your plot. So actually you could not directly compare them. So I cannot really exclude these problems, but what I normally do is I use, for these global analysis, I use like 1 degree grid cells. So that's for the at a global scale, 1 degree latitude, 1 degree longitude. And I just put all the plots together that are found within that larger area, and then I randomly draw individuals out of there to calculate diversity and composition patterns. that is still also very dangerous because some areas have many more plots than others. And also, again, if there is one very big plot and a few small ones, then the big plot will dominate the whole thing. But I did some testing on the influence of plot sizes and plot shapes, and generally there is a pattern. It's around 10 to 20% of the pattern may be biased because of that, but that still leaves quite a lot of variation that you can explain using this kind of method. So there is no perfect solution for this. You just have to do with what you have. You can just try to minimize the problems. So I think if there are many plots all over Indonesia, even though they are all different sizes and different shapes, you can still do a very nice analysis.
Is
my camera.
Have you completed to answer? Okay. How is it, Dr. Ruli? Is that answer your question? Okay, thank you, Professor Slick. The next question is to Dr. Titi Chandra Sunarti from Sylvia Kurniasari, Universitas Erlangga. Thank you for the presentation. My question is why the production of corn flour in this study performed by living cells instead of using crude enzyme, since the product can be utilized by microorganisms and led to reduce the product. Thank you. Please, Dr. Titi.
Yes, thank you to Ibu Sylvia. You are right that the microorganism will reduce the yield of the product, but in the spontaneous and the ordinary process, they use the stepping process, and during the stepping process, a lot of microorganism also grows in the spontaneous fermentation, and it takes a longer time. So if we introduce some microorganism, so it will shorten the fermentation time, so we can save not all the component will solute into the water of the stepping process. So even the microorganism will consume some of the component in the stepping water, but a shortened time will save not all the microorganism can consume the part of the component inside the grids. Okay.
Is that okay, Ibu Sylvia?
Yes, okay, thank you.
Okay, thank you, Ibu Sylvia. The next question is from Izar Khairullah Isari to Dr. Titi. Is there any difference between carbohydrate from roots, crops, and from fruit plants, such as probably from cassava and from rice?
Okay.
Please, Dr. Titi.
Yes. For carbohydrate form in the fruits and also in the roots, of course, it was totally different. Such like in the roots, in the cassava, and also in the sweet potato, yeah, they have a lot of fiber and starch. But in the fruits, such like in ordinary fruits, they a lot of simple sugar and also fiber. But such like wheat and rice, because it was a cereal crops, they not only starch, but also contains some lipid and protein. It becomes different in how to handling this commodity. So the difference between the part of the plant make it different in the carbohydrate composition.
Thank you, Bu Titi. Dr. Pak Izhar, bagaimana? How is the answer? Is that okay with you?
Okay, okay. Thank you for your explanation, Dr. Titi.
Thank you to Dr. Titi. Is there any other question from the audience? We still have about 15 minutes for discussion sessions. While waiting, another question from the audience. Probably I would like to ask a question to Professor Slick. This is just general questions because I'm not an expert in biodiversity. Do you think Southeast Asia countries have enough efforts to prevent biodiversity loss or to manage biodiversity crisis? And how is it compared to other countries in Asia, for example, or other part of the world?
That's a very difficult question.
Oh, I'm sorry.
I think, yeah, all the countries they differ in governments, and but I think overall most of the countries have the capacity to do it, and probably also they have a will to do it. The problem is mostly that it is they have to make a compromise between development and conservation. So the thing is to try to mix the two, where you can both develop and conserve. So that needs a good amount of planning, and also very good and tough law enforcement, which can be very difficult because it differs per country, right? How you can implement that. So I think it's very well possible. There's also already a lot of international collaboration within ASEAN, and we have these big projects like Heart of Borneo, and there was this, I think it's still running, the Greater Mekong project in the Asian mainland. So I think the will is there, and actually I'm still quite positive about the future. Although there are still things going wrong, I think we still have the ability to turn it around.
Okay, thank you very much. And there is still no question from the audience. Probably one more question to Dr. Titi. This is also general questions. There are different kinds of new products, for example, bioethanol for energy and different kinds of polymers for packaging that can be produced from carbohydrates, including starch. Do you think there will be enough starch for food? Because most people, especially people in developing countries, use starch as staple food. What is your suggestion regarding this matter?
There are some competition between the food and energy, especially for the carbohydrate commodity. But we have to find another source for the carbohydrate for the non-food application, because in the global market, the starch source comes from the cassava, especially in the tropical countries. But in the subtropical countries, they use corn and also potato. So in the future, maybe we have to convert the bioenergy not comes from the simple carbohydrates, such like from the sugar or from the starch. So we can go to the next generation of the bioethanol production. Yeah, we can use the residue from the agro-industry, such like solid waste, which can contain a lot of rich in cellulose and also rich in starch, to become the source for the bioethanol production. But don't forget that we have unexplored carbohydrate source, such like sago. It's still plenty of the potential sago in Indonesia also unexplored. So we can use sago not only for food, but also for the non-food application, especially for bioenergy and also for packaging application. Thank you.
Thank you very much, Dr. Titi. Okay, there is some questions from the audience. First one from Ibu Yuni Rahayu from LIPI to the Professor Slick. Do you have a suggestion regarding the planning to move the capital city of Indonesia to East Kalimantan from a conservation and biodiversity perspective? Probably I will read the next question also. Maybe Professor Slick could answer it both together. From Pak Sutomo, how does invasive alien species should be handled in terms of ecosystem health? I think, yeah. There. Okay, Professor Slick, please.
Capital. That's kind of a political
topic. Yeah.
Yeah, I don't know what to think. It seems overall to move a whole capital seems a very difficult thing to do. There's one example already in the world, like, several. Malaysia has tried to set up a new capital city, and there is in Brazil, they set up Brasilia as capital city. I don't think it's going to be very easy to do this, because you have to move all the government agencies there and the people have to follow, and it's a huge infrastructure project. So for the environment in East Kalimantan, it may be a bit risky, but for the economy of Kalimantan, it would be great, of course. So, I don't know exactly. It has to be done in a very, if it's going to happen, it has to be done in a very careful way. It's also a chance, perhaps, if it's going to happen anyway, to show that such a very big move can be done in an environmentally friendly way, right?
Yes.
So there are some opportunities and some dangers, so they have to be considered carefully. For the invasive species, generally, it depends a bit on the invasive species itself. Sorry for my camera again. Some invasive species are not actually that harmful, so they will put.
Aduh, mati. Oh, I'm sorry. Professor Slick, we cannot hear you.
How much did you not hear?
Okay.
The whole answer, not, or just the last part?
The last part only. The last part only.
Oh, okay. Yeah, so there's different kinds of invasive species, and they will need different kinds of management. So the not so dangerous ones are just an addition to the species that are already there. But the dangerous ones, you either have to try to eradicate them from very early on. So then you need a very good monitoring system to detect them. Or you have to create situations, because we have a big problem in Brunei actually with an invasive species, which is Acacia mangium, which is also in Indonesia.
Yeah.
It spreads everywhere where there is enough light. So, and there are now so many, and especially our heath forests are threatened by this species, and they also cause fire, the only way to get rid of them, because it's now almost impossible to remove them by hand, or it's just too expensive and too much work, is to prevent these fires from happening, so that the natural forest can restore itself and outshade these invasive species, because many invasive species are like disturbance species that need a lot of sunlight. So if you can get some kind of canopy cover back, you might also suppress many of these invasive species. So that I think is the most suitable approach for invasive species that have already established and are really degrading the landscape.
Okay, thank you. Professor Slick, how is Ibu Yuni and Pak Sutomo? Is that okay?
It's okay, clear. It's thank you. For
Okay, there is one more question. I think this is the last question because the time is almost up. From Ibu Sylvia Kurniasari, Universitas Erlangga, also to Dr. Titi. My question is, the use of papain enzyme was to reduce the hardness of the substrate. Is it possible if we directly add plant extract that proven contain papain enzyme into the fermentation? And how papain enzyme can reduce the substrate hardness? Thank you. I think this is the last question. Okay.
Okay, thank you, Ibu Sylvia. Actually, if we directly milling the grits to make flour, only around 30% of the flour that we can produce. But addition of the cellulosic enzyme, such as papain in the research, so it will break the hard endosperm, which the protein matrix. So that's why we add the cellulolytic and also proteolytic bacteria. So that's why the hard protein matrix should be break, and it becomes easy for them to milling the hard endosperm of the corn grits.
Okay, how is it, Ibu Sylvia? Is that okay?
it's okay, thank you.
Thank you, Ibu Sylvia. Okay, then I think this is the end of the first plenary sessions. I would like to thank again to Professor Ferry Slick and to Dr. Titi Chandra Sunarti, as well as to all the participants for joining these sessions. I hope you enjoy these sessions, and I give back the chair to Ms. Eva. Ms. Eva, please.
Thank you very much, Professor Ais and the speakers for the informative session. Before proceeding to the next session, I would like to invite all speakers, distinguished guests, and participants to join a photo session. You are pleased to activate your camera. Please be ready. Okay.
Okay.
Thank you very much. To all participants, I would like to remind you to fill the list of attendees via the link provided in the chat. And also, please be aware to assign your username as indicated by the conference rule to enable us to organize you according to your room. For the oral participants, please assign your username using code of room _code of presenter _name. And for non-oral participants, you also need to change your username using code of room _participant _name. Our committee will organize you according to your room. Ladies and gentlemen, we are now having a break session. We will start the parallel session at 1 p.m. We will have a parallel session in two rooms. Room 1 will be hosted by Dr. Renat Ayu Safitri Nurinsyah, and room 2 will be hosted by Dr. Cahyo Rahmadi. At the end of this symposium, we will announce the best presenter from each room. Please join on time. The participants may either stay online or leave the meeting. However, for those who leave the meeting, we require you to join again the symposium at 12:30 using the Zoom link provided by the committee. Thank you and see you in the next session. Ladies and gentlemen, you may have a break session for 1 hour and start our parallel session at 1 p.m. You may either stay online or leave the meeting, but if you leave the meeting, you have to join again to the symposium at 12:30.
Becoming a world-class
Now we have a
institution
break
in
session.
sciences and innovation to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, LIPI. LIPI has various research centers throughout Indonesia under the coordination of the chairman of LIPI, Deputy for Life Sciences, Deputy for Earth Sciences, Deputy for Engineering Sciences, Deputy for Social and Humanities Sciences, and Deputy for Scientific Services. LIPI has a pivotal role in drafting and establishing government regulations and policies related to economic, political, social, and cultural aspects. LIPI also manages scientific data and scientific considerations on biodiversity conservation, as well as the publication of global indexed scientific journals, and also appointed as a scientific authority in coaching national researchers and hosting the inauguration of Indonesian research professors. To support world-class research activities, LIPI managed 5 botanic gardens as a center for ex situ plant conservation, 3 museums that have the biggest collection of flora and fauna specimens in Southeast Asia, and regional level and Indonesian natural history, Barunajaya 8 research vessel, Indonesia Cultural Collection, which keeps the largest collection of microorganisms in Indonesia, and 2 international centers, namely Asia Pacific Center for Ecohydrology, APCE, and Regional Training and Research Center on Marine Biodiversity and Ecosystem Health, RTRC Marbest Center. LIPI has produced qualified research results that are beneficial to the society, including biodiversity, environment and maritime, food security, agriculture and farm, health and pharmacy, advanced material, energy, technology, information and communication, social dynamics, humanity and culture, technology innovation and utilization of science and technology, as well as scientific services, testing and development. LIPI's national scientific collaborations include collaborative research, technology licensing, development, application, and commercialization of research results, non-research collaborations, LIPI's expertise consulting, scientific coaching for youth, students, and teachers with various universities, research institutions, local governments, and industry. As for international collaborations, LIPI conducts joint research, joint laboratories, symposium, workshops, international conferences, exchanges for researchers and experts, as well as international scholarships, while being active as a member and a focal point in various international organizations. LIPI will keep continuing to strengthen national platforms by developing world-class researchers and building scientific collaborations to make great impacts on the nation's competitiveness.
Pulau Enggano, pulau terluar berpenduduk yang secara administratif terletak di Kecamatan Enggano, Kabupaten Bengkulu Utara, Provinsi Bengkulu. Pulau Enggano memiliki panjang 35 km dan lebar 16 km, dengan luas 402,6 km persegi, dengan garis pantai sepanjang 112 km, dengan titik tertinggi di Bukit Koho Bua-Bua, yakni 281 meter di atas permukaan laut, sehingga dapat dikategorikan sebagai pulau kecil. Perpaduan antara pulau terdepan dan pulau kecil membuat Pulau Enggano menjadi sangat penting terkait dengan program pemerintah menyangkut perlindungan dan penyelamatan pulau-pulau kecil dan terdepan negara kesatuan Republik Indonesia. Oceanic Island menjadi keunikan dari Pulau Enggano. Dengan kata lain, Pulau Enggano dalam sejarah geologinya tidak pernah bergabung dengan daratan besar Pulau Sumatera. Kecamatan Enggano terdiri dari 6 desa yang letaknya di pinggir pantai dan berbatasan langsung dengan Samudra Hindia. Keenam desa tersebut adalah Desa Banjarsari, Desa Meok, Desa Apoho, Desa Malakoni, Desa Kaana, dan Desa Kahyapu dengan pusat pemerintahan di Desa Apoho. Dari segi sejarah, Pulau Enggano pernah menjadi salah satu tempat transit jalur perdagangan internasional pada masa awal kolonialisasi di Nusantara. Hal ini menyebabkan Pulau Enggano memiliki keunikan tersendiri dari segi bahasa, tradisi, dan budayanya. Masyarakat Pulau Enggano terdiri dari 6 suku, yaitu suku Kauno, suku Kaarubi, suku Kaitora, suku Kaharuba, suku Kahua, dan suku Kamai. Mereka menganut sistem matrilineal atau mengikuti garis ibu dalam hal keturunan dan suku. Nama Enggano pertama kali dituliskan oleh Cornelis de Houtman pada saat melakukan ekspedisinya bersama 4 kapal ekspedisi yang bernama Mauritius, Hollandia, Amsterdam, dan Duyfken. Pada catatannya, Houtman menuliskan 0506 1596 tiba di Pulau Hindia yang pertama, Pulau Enggano, di sebelah barat Pulau Sumatera bagian selatan. Pada tahun 1771, Charles Miller berhasil berlabuh dan bertemu penduduk asli Pulau Enggano. 100 tahun kemudian, tepatnya di tahun 1865, Mr. Francis datang ke Pulau Enggano dan mendapatkan kesan yang mendalam dari penduduk Pulau Enggano. Hal tersebut diketahui oleh seorang linguistik asal Jerman, Hans Kehler, yang mengunjungi Pulau Enggano pada tahun 1930-an. Adalah kemudian seorang pengelana dunia asal Italia yang bernama Elio Modigliani, yang kemudian mencatatkan Enggano dalam sejarah melalui bukunya L'Isola delle Donne, Viaggio ad Enggano. Elio Modigliani berlabuh di Pulau Enggano pada tahun 1891 dan tinggal di sana selama 8 bulan, yang kemudian mencatatkan temuan keanekaragaman fauna di Pulau Enggano. Di tahun 1936, naturalis asal Belanda, Wilhelm Jan Lutjeham dan Jocelyn de Jong melakukan ekspedisi dan koleksi flora dan fauna di Pulau Enggano. Dan di tahun 2015, Kedeputian Ilmu Pengetahuan Hayati, Lembaga Ilmu Pengetahuan Indonesia, melakukan ekspedisi Widya Nusantara untuk meneliti keanekaragaman hayati, menemukan potensi bioaktif dari taksa terpilih yang dapat dikembangkan sebagai bahan pangan, obat-obatan, dan sumber energi. Kegiatan penelitian ini dirancang bersinergi dengan berbagai aspek, mulai dari eksplorasi, identifikasi, hingga analisis potensi dan pemanfaatan.
Enggano dipilih sebagai prioritas. Jadi kalau di Berkepribadian Ilmu Pengetahuan Hayati LIPI itu ada beberapa program unggulan LIPI, dan salah satunya adalah eksplorasi bioresources di Pulau Enggano. Jadi ini merupakan salah satu kegiatan unggulan Berkepribadian Ilmu Pengetahuan Hayati. Kenapa dipilih Enggano? Itu ada beberapa pertimbangan. Pertama adalah pertimbangan saintifik, itu karena pulau ini adalah oceanic island, di mana pulau ini dalam sejarah geologinya itu tidak pernah bergabung dengan Pulau Sumatera. Nah, konsekuensinya itu jenis-jenis atau spesiasi yang ada di pulau ini tetap unik, banyak jenis-jenis endemik, dan yang paling mendasar adalah informasi tentang jenis-jenis itu belum banyak. Jadi belum ada data-data yang lengkap mengenai jenis-jenis yang ada di pulau tersebut. Yang kedua, secara geopolitik, pulau ini merupakan salah satu pulau terluar di kawasan Nusantara, di mana pulau-pulau kawasan terluar itu menjadi prioritas pembangunan, sehingga untuk melaksanakan pembangunan itu harus ada data-data yang mendukung. Data-data yang mendukung adalah data dasar potensi dari pulau tersebut, termasuk adalah eksplorasi potensi bioresources Pulau Enggano.
Kegiatan ekspedisi Pulau Enggano dilakukan oleh para peneliti dari Pusat Penelitian Biologi, Pusat Penelitian Bioteknologi, Pusat Konservasi Tumbuhan Kebun Raya Indonesia, dan Pusat Penelitian Biomaterial. Kegiatan ekspedisi bertujuan mendata jenis-jenis flora, fauna, dan mikroorganisme yang ada di Pulau Enggano, salah satu pulau terdepan di Provinsi Bengkulu. Dengan keunikan sejarah geologinya, Pulau Enggano diyakini memiliki tingkat endemisitas flora dan fauna yang tinggi. Pulau Enggano memiliki ekosistem khas pulau kecil, ekosistem alami seperti ekosistem laut, ekosistem semi-terestrial seperti mangrove dan tepi sungai, serta ekosistem terestrial, yakni hutan pantai, hutan dataran rendah, dan rawa. Ekosistem hutan pantai dapat ditemukan hampir di sepanjang garis pantai timur Pulau Enggano. Karakteristik pantai yang terdapat di Pulau Enggano adalah pasir berlumpur, pasir berkarang, pasir karang berlumpur, dan pantai karang berbatu yang keberadaannya berkaitan erat dengan ekosistem terumbu karang dan mangrove. Peneliti dari Pusat Penelitian Bioteknologi dan Pusat Penelitian Biologi mengeksplorasi dan mengumpulkan mikroorganisme dari air laut, sedimen laut, pasir laut, spons, tanah, serasah, daun, bunga, buah, jamur endofitik, dan jaringan atau organ hewan. Koleksi mikroorganisme ini dilakukan untuk mencari agen pembentukan senyawa obat, penghasil energi, dan penghasil enzim penting dalam sektor pangan dan pengolahan produk pangan. Dalam ekspedisi ini, para peneliti telah berhasil mengkoleksi mikroorganisme antara lain Actinobacteria, bakteria, khamir, jamur, arkea, dan alga. Indonesia telah dikaruniai ragam hutan alam yang terbentang dari ujung barat di Sabang sampai ujung timur di Merauke. yang merupakan habitat utama berbagai jenis flora dan fauna. Hutan pantai merupakan bagian dari ekosistem pesisir dan laut yang menyediakan sumber daya alam yang produktif, baik sebagai sumber pangan, penghasil obat-obatan yang bernilai ekonomi tinggi, penemuan produk kimia, tambang mineral, dan sumber energi, serta kawasan rekreasi atau pariwisata pantai. Pulau Enggano memiliki hutan mangrove paling luas di Provinsi Bengkulu, yakni 1.710 hektar. Tanjung Kaana merupakan daerah yang mempunyai hutan mangrove paling lebat. Selain hutan mangrove, Pulau Enggano juga memiliki daerah rawa dengan luas 1.967 hektar. Pada kedua ekosistem ini, ditemukan mikroorganisme yang dapat hidup dalam kondisi anaerob atau kondisi tanpa oksigen. Ekosistem mangrove banyak ditemukan di Pulau Enggano. Jenis dominan seperti Bruguera gymnorrhiza yang dapat ditemukan di sisi tenggara dan barat daya pulau. Hutan mangrove yang tumbuh di pantai daerah tropis merupakan habitat bagi berbagai hewan. Burung, mamalia, dan reptil mendapatkan keuntungan dari kayanya pasokan makanan di wilayah ini seperti ikan, kerang, tiram, siput, anemon laut, dan kepiting. Selain itu, hutan mangrove melindungi pantai dari terjangan gelombang dan banjir. Di Pulau Enggano mengalir beberapa sungai besar dan kecil yang memiliki hulu di bagian tengah pulau. Kita dapat menemukan jenis tumbuhan seperti Nipa frutican atau nipah, Sakarum atau gelagah, Pometia pinnata atau matoa, serta pada daerah sungai yang dekat dengan tepi laut dapat ditemukan Terminalia catapa atau ketapang, dan Barringtonia macrocarpa atau putat. Di Pulau Enggano ditemukan jenis tumbuhan berkayu potensial yang telah dimanfaatkan dan memiliki potensi ekonomi seperti merbau, nehek, ketapang, dan apua. Selain itu juga ditemukan daun pandan sebagai sumber serat alam untuk pembuatan papan serat. Rayap merupakan serangga yang memiliki peranan penting di dalam ekosistem hutan. Di Pulau Enggano, rayap sangat mudah dijumpai sepanjang tahun dan memiliki kasta steril dengan subfamili Macrotermitine, Nasutitermitine, dan Rhinotermitine. Kekayaan jenis burung endemik dari kawasan Pulau Enggano jelas terlihat dan bahkan tidak menutup kemungkinan akan adanya temuan jenis endemik yang lain. Pulau Enggano juga dikategorikan sebagai endemic bird area oleh BirdLife International karena memiliki keanekaragaman jenis burung yang sangat menarik. Betet ekor panjang, anis kembang Enggano, uncal buau, kacamata Enggano, dan celepuk Enggano dapat ditemui di pulau ini. Koleksi ilmiah dilakukan dengan memperhatikan aspek konservasi dan tidak mengganggu keseimbangan populasi di alam. Dalam ekspedisi ini dijumpai sebanyak 33 jenis burung di Pulau Enggano. Selama ekspedisi, para peneliti ini menemukan mamalia kecil, antara lain 11 jenis Chiroptera atau kelelawar dan 2 jenis Rodentia. Selain itu juga diperoleh informasi tentang beberapa mamalia besar seperti babi hutan, sapi liar, kerbau liar, dan musang luwak. Babi hutan merupakan jenis paling dominan dan terdistribusi mulai dari dataran rendah, hutan rawa, sampai hutan pantai. Keunikan herpetofauna di pulau yang terletak di Samudra Hindia ini telah diketahui sejak munculnya publikasi mengenai jenis-jenis herpetofauna Pulau Enggano pada abad ke-19. Pada ekspedisi ini, ditemukan 13 jenis reptil dan 2 amfibi di Pulau Enggano, seperti ular tikus Enggano atau Colognathus engganensis, dan cicak terbang atau Dracomodigliani. 3 jenis lainnya yang dijumpai adalah kura-kura batok, ular sanca batik, dan biawak. Selain itu, buaya muara juga teramati di sepanjang Sungai Kuala Besar. Serangga yang dikoleksi selama ekspedisi adalah kupu-kupu, ngengat, capung, dan lalat buah. Secara umum, jenis serangga yang dijumpai di Pulau Enggano tidak terlalu beragam bila dibandingkan dengan pulau-pulau kecil lainnya. Namun demikian, Pulau Enggano memiliki jenis serangga yang endemik seperti ngengat Enggano atau Pilodes engganensis, dan capung jarum Enggano atau Pseudogreyon Philidorsum engganoense. Ada 24 jenis moluska atau keong-keongan yang terdiri dari 17 jenis moluska darat dan 7 moluska air tawar dijumpai selama ekspedisi. Jumlah ini sedikit lebih banyak daripada jumlah jenis yang dijumpai sebelumnya. Jenis-jenis krustase yang dijumpai selama ekspedisi ini sebanyak 25 jenis. 1 jenis diantaranya merupakan catatan baru, yaitu kepiting atau Discoplak magna. Selain itu, 2 jenis udang dimungkinkan merupakan kandidat jenis baru. Dari eksplorasi flora di Pulau Enggano, diperoleh sebanyak 275 jenis. Semua koleksi merupakan jenis tumbuhan tinggi yang meliputi kelompok gymnosperm, monokotil dan dikotil yang diperoleh dari tumbuhan dengan habitus pohon kecil, perdu, herba, liana, maupun epifit. Dari keseluruhan koleksi, terdapat 5 jenis endemik yang berhasil ditemukan, yaitu Isora engganensis, Polysias engganensis, Medinilla engganensis, Barringtonia revoluta, dan Diospheros scabiosa. Zingiberaceae ditemukan hanya sedikit di Pulau Enggano. Habitat yang terlalu panas dan kurang lembab kemungkinan menjadi salah satu faktor tidak banyak ditemukannya Zingiberaceae. Demikian halnya dengan Begoniaceae dan Gesneriaceae yang memiliki potensi sebagai tanaman hias. Flora palem dominan ditemui adalah Onchospermatigillarium atau nibung, serta Pandanus dubius dan Pandanus tectorius untuk pandan. Kedua jenis pandan ini ditemukan hidup berdampingan. Jamur yang dikoleksi di Pulau Engganu, yaitu Basidiomycota dan Ascomycota. Koleksi jamur makroskopis yang berhasil dikumpulkan tergolong dalam kelompok Auriculariaceae, Agaricales, Ganodermataceae, Polyporaceae, dan Sarcosipiceae. Sedangkan koleksi lumut dilakukan di berbagai substrat di tempat-tempat lembab atau basah. Lumut yang ditemukan di lokasi penelitian sebagian besar bersubstrat batang pohon, sedangkan di tanah dan batu sangat jarang ditemukan. Hutan dataran rendah yang masih merupakan hutan primer masih dapat ditemui di bagian tengah pulau. Sedangkan hutan dataran rendah yang digolongkan sebagai hutan sekunder banyak ditemukan pada bagian utara pulau, di mana pengaruh antropogenik paling banyak ditemukan. Banyak tumbuhan berkhasiat obat yang tumbuh di Pulau Engganu. Selain itu ada pula komoditas pertanian yang utama di Pulau Engganu, yakni budidaya pisang, coklat, kedondong, dan salak yang tumbuh liar di hutan. Beberapa komoditas pangan yang belum maksimal terungkap di Pulau Engganu adalah buah pandan dan buah nipah. Tumbuhan paku banyak ditemui di Pulau Engganu. Selain itu, suku talas-talasan dan anggrek juga menjadi jenis kekayaan hayati yang akan diaklimatisasi dan ditanam sebagai koleksi kebun raya Indonesia. Riparian atau hutan tepi sungai yang ditemukan di daerah Koala Besar didominasi oleh Calophyllum inophyllum atau nyamplung dan Stemonurus. Jenis lain yang ditemukan di sekitar sungai adalah jenis-jenis tumbuhan yang umum ditemukan di tepi pantai, seperti Pomesia pinnata, Terminalia catapa, dan Hibiscus teleaceus atau waru laut. Pulau Engganu merupakan salah satu pulau terdepan di Indonesia yang belum terungkap keanekaragaman iktiofaunanya. Sebanyak 51 jenis ikan ditemukan di Pulau Engganu. Satu jenis diantaranya yaitu Stipodon sp, diduga merupakan jenis baru. Sedangkan 5 jenis yang lain perlu dikaji lebih lanjut untuk mengetahui status taksonominya. Antara lain, jenis-jenis dari anggota marga Hypseleotris, Redigobius, Stenogobius, Sismatogobius, Ambasis, dan Mugilogobius. Tidak tertutup kemungkinan bila jenis-jenis ini merupakan spesies endemik di Pulau Engganu. Hutan lindung Koho Buah-Buah seluas 3.450 hektar berada di puncak tertinggi Pulau Engganu, yakni 240 meter di atas permukaan laut yang merupakan hulu dari sungai Koala Besar dan Koala Kecil di Malakoni. Hutan lindung Koho Buah-Buah merupakan area berbukit-bukit dengan puncak tertinggi mencapai 281 meter yang sekaligus titik tertinggi di Pulau Enggano. Selain melakukan pengkoleksian, tim ekspedisi Pulau Enggano juga melakukan penelitian untuk mendapatkan bahan pangan sumber karbohidrat berbahan umbi dan serealia lokal, serta bibit tanaman, buah-buahan, dan nira sebagai sumber bakteri asam laktat yang dapat digunakan untuk meningkatkan kualitas produk. Ekosistem Pulau Enggano kini mengalami kerusakan yang cukup berarti akibat berbagai aktivitas manusia. Yang mengkhawatirkan adalah beberapa kawasan hutan tersebut telah menjadi milik perorangan, akan dibukanya lahan transmigrasi nelayan, dan akan dibuatnya jalan Outer Ring Road yang akan membuka kawasan-kawasan hutan pantai tersebut. Perubahan penggunaan lahan dapat dilihat dengan jelas melalui hasil interpretasi citra Landsat yang diambil dari tahun ke tahun. Tidak berbeda dengan daerah lain di Indonesia, pengaruh perilaku kehidupan dan tata cara hidup modern secara perlahan menenggelamkan kearifan lokal yang ada di Pulau Enggano sejak dulu. Dalam ekspedisi ini, para peneliti etnobotani melakukan penelitian mengenai perubahan lahan di Pulau Enggano berdasarkan pengetahuan lokal masyarakat, konservasi alam, dan persepsi terhadap lingkungannya. Melalui kegiatan Participatory Rural Appraisal, masyarakat Enggano juga diminta untuk membuat sketsa desa di masa lalu, sekarang, dan perencanaan di masa depan. Terlihat dalam sketsa tersebut, pada masa awal hingga tahun 1883, terlihat masyarakat Enggano masih tinggal di pedalaman dan menetap di Koho Buah-Buah. Pada masa transisi di tahun 1883 hingga 1950, lahan perkebunan sudah mulai bernilai komersil. Masyarakat mulai menanam dan menjual hasil bumi. Dan pada masa 1950 hingga kini, secara perlahan para pemukim makin mendekat bergerak ke pantai, yaitu ke arah sumber daya dan pasar. Pemanfaatan tumbuhan dan usaha pelestarian yang dilaksanakan oleh masyarakat tiap wilayah atau daerah akan berbeda, tergantung pada ekosistem dan waktu. Seperti masyarakat Enggano, antara satu generasi dengan generasi berikutnya memiliki pengetahuan dan pemanfaatan yang berbeda. Walaupun demikian, ada beberapa pemanfaatan tumbuhan yang masih tetap dipertahankan oleh masyarakat, seperti pembuatan alat musik dari batok kelapa, ukulele dari kayu bakbak, pemanfaatan tumbuhan untuk obat-obatan seperti kumis kucing, temulawak, ataupun sumber daya alam pangan seperti ganyong.
Banyak hasil yang kami dapat, terutama sekali dari mikrobiologi misalnya, karena sama sekali tidak ada data tentang flora maksud saya biota mikro.
Memang tidak terkenal seperti Pulau Dewata, Bali, yang banyak dikunjungi wisatawan lokal dan mancanegara. Namun keindahan pantai di pulau terpencil ini juga menyimpan beribu potensi wisata maupun sumber ekonomi seperti udang lobster. Selain lobster, Simelu juga terkenal dengan penghasil cengkeh yang menjadi komoditi ekspor. Bagi para peselancar, Pulau Simelu adalah surga. Ombak dengan ketinggian 5 meter, laut bersih, dan langit cerah menjadi idaman banyak peselancar. Apalagi Simelu belum banyak dikunjungi orang, sehingga membuat peselancar leluasa menyalurkan hobinya. Apakah Pulau Simelu juga surga bagi keanekaragaman hayati Indonesia? Kami melakukan pembuatan plot permanen dengan menggunakan petak untuk mengungkap kondisi tegakan vegetasi. Selain itu, kami juga melakukan eksplorasi dan menginventarisasi tumbuhan dengan cara sampling pada setiap lokasi yang menggunakan sistem lahan didasarkan pada asumsi keanekaragaman flora yang dipengaruhi oleh kondisi habitat yang berbeda, mencakup topografi, bahan induk, dan jenis tanah. Kecuali untuk jenis anggrek. Kami menyusuri pantai karena banyak dijumpai jenis-jenis anggrek di tempat terbuka sepanjang pantai. Berbagai jenis pandan yang terkenal dengan sumber serat alam juga kami temukan di sini. Kami menyusuri lokasi dan melakukan koleksi semua jenis tumbuhan lumut yang memiliki generasi gametofit dan sporofit. Kami melakukan survei kondisi sosial dan lingkungan di Pulau Simelu. Pulau Simelu yang pada gempa bumi dan tsunami tahun 2004 silam memakan korban jiwa sangat minim karena smog. Ya, smog merupakan pelajaran hidup bagi masyarakat Simelu. Melalui adat tutur, kearifan lokal, cerita turun-menurun, membuat masyarakat Simelu selalu siap siaga menghadapi gempa bumi dan tsunami. Berkilo-kilo jarak menyusuri jalan setapak yang lengang, diterpa angin bersama hangatnya matahari, kami tidak hanya melakukan koleksi flora, tetapi juga fauna. Kami melakukan koleksi lalat buah dengan perangkap senyawa atraktan. Melalui pengamatan metode jalur dan penangkapan menggunakan jaring kabut, kami melakukan koleksi jenis burung. Sedangkan penelitian mamalia dilakukan dengan menggunakan kombinasi penangkapan, yaitu dengan perangkap dan pengamatan langsung. Pulau Simelu merupakan salah satu pulau terdepan di Indonesia yang belum terungkap keanekaragaman iktiofaunanya. Dengan beberapa jenis alat tangkap, kami melakukan koleksi iktiofauna. Kami mengumpulkan berbagai sampel mikroorganisme untuk mencari agen pembentukan senyawa obat, penghasil energi, dan penghasil enzim penting dalam sektor pangan dan pengolahan produk pangan. Selama melakukan ekspedisi di Pulau Simelu, kami juga melakukan sampling riap kayu, jamur, rayap, tanah, stalaktit, stalagmit, dan lumpur goa, serta serat alam. Seluruh kegiatan tersebut kami lakukan untuk mengungkap kekayaan Indonesia di pulau terdepan, memperkaya ketersediaan informasi sehingga dapat membantu dalam pengelolaan kegiatan kawasan atau diharapkan menjadi informasi tambahan yang berguna sebagai ilmu pengetahuan. Kami berharap potensi sumber daya alam yang ada dapat dikembangkan untuk mendukung pembangunan daerah serta meningkatkan kesejahteraan masyarakat. Pemanfaatan dan usaha pelestarian yang dilakukan oleh masyarakat tiap wilayah atau daerah akan berbeda. Kekayaan hayati Pulau Simelu memiliki potensi untuk dikembangkan dan dimanfaatkan. Selama 14 hari, kami melewati hari-hari dengan teriknya matahari, hujan deras yang datang tiba-tiba, dan medan hutan yang penuh dengan tantangan. Pada ekspedisi Bioresources Pulau Simelu ini, kami berhasil mengungkap berbagai jenis kekayaan flora dan fauna, potensi varietas tanaman pangan lokal untuk diversifikasi pangan, isolate mikroorganisme, serta potensi keanekaragaman hayati lainnya. Karena kami meyakini bahwa hidup adalah petualangan dan tantangan. Kami meyakini bahwa hidup adalah tentang membuat perubahan. Dan kami membuat perubahan melalui riset, melewati seluruh tantangan untuk hasil terbaik bagi nusa dan bangsa. Ekspedisi Bioresources Indonesia merupakan salah satu bentuk dari upaya LIPI untuk mengintervensi, mengkoleksi, mengkonservasi, sekaligus melakukan penelitian terhadap potensi keanekaragaman hayati, khususnya di wilayah pulau-pulau kecil terdepan. Pulau-pulau kecil menjadi fokus utama karena selama ini keanekaragaman hayati di wilayah-wilayah ini memang belum pernah dieksplorasi. Dan sebagai negara kepulauan, Indonesia memiliki ribuan pulau-pulau kecil sebagai salah satu aset nasional yang harus dikelola dengan sebaik mungkin. Setelah tahun 2015 melakukan ekspedisi ke Pulau Enggano dan ekspedisi ke Pulau Sumba di tahun 2016, pada tahun 2017 kami, para peneliti di Lembaga Ilmu Pengetahuan Indonesia, melakukan perjalanan ke Pulau Simelu, pulau yang terletak di sebelah barat Provinsi Aceh dan berada di Samudra Hindia. Bertolak dari Jakarta, kami, tim ekspedisi, menggunakan pesawat terbang menuju Medan dan berlanjut terbang menuju Sinabak, waktu tempuh Medan Simelu saat cuaca cerah 1 jam 10 menit. Pulau Simelu memang tidak terkenal seperti Pulau Dewata, Bali, yang banyak dikunjungi wisatawan lokal dan mancanegara. Namun keindahan pantai di pulau terpencil ini juga menyimpan beribu potensi wisata maupun sumber ekonomi seperti udang lobster. Selain lobster, Simelu juga terkenal dengan penghasil cengkeh yang menjadi komoditi ekspor. Bagi para peselancar, Pulau Simelu adalah surga. Ombak dengan ketinggian 5 meter, laut bersih, dan langit cerah menjadi idaman banyak peselancar. Apalagi Simelu belum banyak dikunjungi orang, sehingga membuat peselancar leluasa menyalurkan hobinya. Apakah Pulau Simelu juga surga bagi keanekaragaman hayati Indonesia? Kami melakukan pembuatan plot permanen dengan menggunakan petak untuk mengungkap kondisi tegakan vegetasi. Selain itu, kami juga melakukan eksplorasi dan menginventarisasi tumbuhan dengan cara sampling pada setiap lokasi yang menggunakan sistem lahan didasarkan pada asumsi keanekaragaman flora yang dipengaruhi oleh kondisi habitat yang berbeda, mencakup topografi, bahan induk, dan jenis tanah. Kecuali untuk jenis anggrek. Kami menyusuri pantai karena banyak dijumpai jenis-jenis anggrek di tempat terbuka sepanjang pantai. Berbagai jenis pandan yang terkenal dengan sumber serat alam juga kami temukan di sini. Kami menyusuri lokasi dan melakukan koleksi semua jenis tumbuhan lumut yang memiliki generasi gametofit dan sporofit. Kami melakukan survei kondisi sosial dan lingkungan di Pulau Simelu. Pulau Simelu yang pada gempa bumi dan tsunami tahun 2004 silam memakan korban jiwa sangat minim karena smog. Ya, smog merupakan pelajaran hidup bagi masyarakat Simelu. Melalui adat tutur, kearifan lokal, cerita turun-menurun, membuat masyarakat Simelu selalu siap siaga menghadapi gempa bumi dan tsunami. Berkilo-kilo jarak menyusuri jalan setapak yang lengang, diterpa angin bersama hangatnya matahari, kami tidak hanya melakukan koleksi flora, tetapi juga fauna. Kami melakukan koleksi lalat buah dengan perangkap senyawa atraktan. Melalui pengamatan metode jalur dan penangkapan menggunakan jaring kabut, kami melakukan koleksi jenis burung. Sedangkan penelitian mamalia dilakukan dengan menggunakan kombinasi penangkapan, yaitu dengan perangkap dan pengamatan langsung. Pulau Simelu merupakan salah satu pulau terdepan di Indonesia yang belum terungkap keanekaragaman iktiofaunanya. Dengan beberapa jenis alat tangkap, kami melakukan koleksi iktiofauna. kami mengumpulkan berbagai sampel mikroorganisme untuk mencari agen pembentukan senyawa obat, penghasil energi, dan penghasil enzim penting dalam sektor pangan dan pengolahan produk pangan. Selama melakukan ekspedisi di Pulau Simelu, kami juga melakukan sampling riap kayu, jamur,
menjelajah Kepulauan Tanimbar sama dengan menjelajah budaya dan tradisinya. Di beberapa bagian lain Kepulauan Tanimbar, bahkan mampu memanjakan mata dengan pemandangan alam yang menakjubkan. Sebagian besar mata pencaharian masyarakat ini adalah berladang dan menangkap ikan untuk menjala rupiah. Sedangkan para wanita membantu dengan menenun agar asap dapur tetap mengepul. Tidak hanya untuk dijual, hasil tenunan juga akan digunakan untuk mereka dan kerabatnya. Sejumlah masyarakat memiliki bakat seni dan pengetahuan yang diperoleh secara turun-temurun untuk membuat kerajinan. Kayu eboni atau kayu arang digunakan sebagai bahan untuk membuat ikat pinggang, replika kapal, patung, dan lain-lain. Sejak masuknya Kristen, sebagian besar masyarakat memeluk agama Kristen dan Katolik. Walaupun begitu, kepercayaan tradisional terhadap leluhur atau nenek moyang masih tetap kuat dipegang. Perpaduan antara kepercayaan lokal dan agama Katolik kentara jelas tatkala kami datang ke sana. Permohonan izin kepada Tuhan dan leluhur dihaturkan supaya tetap dalam lindungannya.
Untuk kelancaran, kemudian dan terutama juga keselamatan kami, karena terus terang kami pendatang, kami mungkin ingin tahu.
Para tua-tua adat menggunakan doa Bapa Kami dan Salam Maria dalam sembahyang tersebut. Dalam doa yang dihaturkan, biasanya teriring sopi yang menjadi simbol lokal dari persaudaraan, tanda upacara, ritual, bahkan pemecah permasalahan. Sopi menjadi hal yang wajib jika ada kegiatan yang berkaitan dengan adat. Sopi merupakan media antara tuan tanah ataupun tua-tua adat untuk berkomunikasi dengan moyang dan leluhur. Selain kaya akan budaya dan tradisinya, Kepulauan Tanimbar dipercaya menyimpan keanekaragaman hayati yang kaya akan endemisitas karena letak geografisnya berada di antara Kepulauan Maluku di utara, Sulawesi di barat laut, Kepulauan Sunda Kecil di barat, Australia di selatan, dan Papua di timur. Baru saja kami menikmati kota Saum Laki di Pulau Yamdena ketika menerima kabar bahwa Balai Konservasi Sumber Daya Alam telah melakukan penyitaan burung kakatua goviniana atau kakatua Tanimbar yang berasal dari masyarakat setempat. Burung ini merupakan burung asli dan endemik di Kepulauan Tanimbar yang dilindungi dengan PP Menteri Lingkungan Hidup dan Kehutanan Republik Indonesia nomor P.MEN-LHK-Kum.1-1-8-2018 tentang perubahan atas Peraturan Menteri Lingkungan Hidup dan Kehutanan nomor P.2.-MEN-LHK-Kum.1-6-2018 tentang jenis tumbuhan dan satwa yang dilindungi. Dalam hal status perdagangan yang mengacu pada Convention on International Trade in Endangered Species of Wild Fauna and Flora atau CITES dan pada International Union for Conservation of Nature atau IUCN, status kakatua Tanimbar berstatus terancam. Berbagai jenis burung kelompok Psittacidae dan Kakatuidae, termasuk diantaranya kakatua Tanimbar, merupakan komoditi perdagangan untuk ekspor keluar wilayah Indonesia guna memenuhi kesenangan manusia.
Kasus penyelundupan begini ya jadi ancaman juga bagi satwa. Nah itu yang mungkin yang lebih fatal lagi kalau yang baru ditangkap dari alam. Karena apapun dia kan masih liar. Jadi dipaksain kan dia pasti melawan. Tapi kalau yang sudah dipelihara lama agak jinak pasti dia ikut nurut tuannya kan. Nah seperti itu. Ya seperti ini. Ini cara-cara yang mereka lakukan untuk penyelundupan satwa antar pulau kan. Ya. Jadi bisa satwa ini burung dia dimasukin di sini, ya. Terus mereka ya kelabui petugas, masukin dalam tas. Ya. Baru dia. Jadi kita susah untuk. Nah, itu yang kasus-kasus yang pernah kami alami itu. Akhirnya kan mati semua. Pelakunya kami tangkap, proses, ya satuannya kami amankan di PRS, akhirnya satuannya juga mati. Nah seperti itu. Yang disayangkan juga ini kasus seperti ini akan terjadi di Tanimbar seperti ini yang sekarang ini. Pergi dan berkembang biak kau.
Peneliti avifauna Tri Haryoko bersama rekan-rekan peneliti lain bergegas menuju lokasi penyitaan dan kemudian membawa 56 kakatua Tanimbar ke kandang habituasi. Tri pernah ke Pulau Tanimbar dan melakukan penelitian kerjasama berfokus kepada spesies kakatua Tanimbar. Penelitian kerjasama tersebutlah yang kemudian membawa Tri kembali ke Kepulauan Tanimbar bersama rekan-rekan peneliti dari Kedeputian Ilmu Pengetahuan Hayati, Lembaga Ilmu Pengetahuan Indonesia. Tri melakukan pengamatan dan identifikasi di beberapa titik di Kepulauan Tanimbar, yakni Pulau Yamdena, Pulau Selaru, dan Pulau Larat. Kakatua Tanimbar merupakan burung paruh bengkok asli Indonesia. Spesies ini merupakan burung endemik di Kepulauan Tanimbar yang berada di Provinsi Maluku.
Sebagai burung yang endemik di Kepulauan Tanimbar, ini sangat menarik perhatian peneliti ataupun wisatawan untuk melakukan bird watching. Karena memang burung ini hanya ada di Kepulauan Tanimbar. Kita masih dapat mudah menjumpai burung-burung tersebut di area ladang maupun pinggir hutan di sekitar Pulau Yamdena, Pulau Selaru, Pulau Larat. Kita masih mudah menjumpainya. Walaupun secara populasi tidak sebanyak tahun 80-an yang diperkirakan populasi sekitar 300 sampai 400 ribu individu.
Kakatua Tanimbar memiliki kecerdasan yang luar biasa. Burung ini telah mampu memecahkan permasalahan kakatua Tanimbar dalam mendapatkan makan, seperti halnya yang dilakukan oleh kelompok hewan primata. Berbagai penelitian dilakukan untuk mengetahui tingkat kecerdasan di alam maupun di laboratorium. Kakatua Tanimbar memiliki bulu-bulu berwarna putih, kekang merah jambu, penutup telinga semu kekuningan, dengan dada dan tubuh bagian bawah berwarna merah muda. Bulu terbang di bagian bawah dan bulu ekor berwarna kekuningan. Paruh abu-abu atau putih. Pada burung jantan, mata berwarna coklat gelap. Tetapi pada burung betina, mata berwarna coklat kemerahan. Ini bisa dijadikan cara efektif untuk membedakan jenis kelaminnya. Burung kakatua ini memiliki suara parau namun mampu memaki keras. Panjang tubuhnya sekitar 31 sampai 32 cm dengan bobot badan rata-rata 350 gram. Inilah spesies terkecil dari semua keluarga Kakatuidae. Burung kakatua goviniana atau kakatua Tanimbar ini memasuki masa bereproduksi atau breeding antara bulan Agustus atau Desember dengan jumlah telur 1-2 butir telur. Namun demikian, dari penelitian yang kita lakukan, ketika kita melakukan pengecekan pada masa anakan, kita hanya menjumpai 1 ekor saja anakan yang ada di dalam lubang sarang. Jadi, tidak semua telur menetas menjadi anakan atau tetap hidup sampai dia terbang sendiri. Matahari belum begitu tinggi ketika Tri memasuki pinggiran hutan di desa Lorulun. Keragaman jenis burung yang dimiliki Kepulauan Tanimbar memiliki potensi untuk dapat dijadikan sebagai situs pengamatan burung atau bird watching yang semakin ramai digandrungi oleh wisatawan sebagai wisata minat khusus. Tidak hanya kakatua Tanimbar saja yang dapat dijumpai di Kepulauan Tanimbar, tetapi ada beberapa jenis burung lainnya yang dapat kita jumpai. Ada jenis burung nuri yang juga endemik, beberapa jenis elang yang bisa dijumpai di pinggir hutan, di dekat ladang, maupun di dalam hutan. Beberapa jenis kelompok merpati-merpatian dari yang kecil hingga besar juga mudah dijumpai di sana. Beberapa jenis burung air juga dapat mudah dijumpai, terutama di rawa. Selain beberapa jenis burung penetap, rawa juga merupakan salah satu tempat burung-burung bermigrasi dari Australia. Sudah beberapa hari lamanya kakatua tanimbar berada di kandang habituasi dan siap untuk dilepaskan kembali ke alam. Satu persatu kakatua tanimbar diberikan cincin untuk mengetahui identitas burung tersebut dan memudahkan monitoring.
Sebelum burung itu dilepaskan kembali ke alam, kita harus lakukan perlakuan terlebih dahulu terhadap burung kakatua goviniana ini. Oleh karena itu ketika melakukan penyitaan, burung-burung itu harus dihabituasi dulu, diadaptasikan untuk bisa menjadi liar kembali sebelum kita lepaskan. Nah, dengan proses habituasi ini diharapkan burung akan menjadi liar kembali. Jadi setelah proses habituasi, sebelum dilepas liarkan dengan metode pencincinan. Jadi setiap individu yang akan dilepas liarkan, kita kasih tanda cincin. Cincin kita taruh di kaki sebelah kanan, karena kaki kiri ini menjadi kebiasaan burung itu ketika dia makan, megang pakan itu ada di kaki kiri, sehingga cincin tadi kita taruh di kaki kanan.
Hari demi hari berlalu, dan di suatu sore yang hangat, kami berkumpul bersama di Pulau Anggarmasa, bagian dari Kepulauan Tanimbar. Bersama para ibu yang tengah berkumpul, bersenandung bersama, yang menjadi ciri khas keramah tamahan pulau ini. Mereka menyenandungkan lagu tentang keindahan Pulau Tanimbar dan keramahan masyarakatnya. Matahari perlahan terbenam di ufuk barat, diganti dengan kegelapan malam. Ada beberapa cerita rakyat yang menceritakan asal-muasal kakatua Tanimbar. Masyarakat Tanimbar menyebutnya tilngoy lempitenan.
Manik tilngoy itu bahasa Tanimbar ya. Kalau burung kakatua ya bahasa Indonesia. Jadi manik burung, tilngoy kakatua.
Salah satu cerita rakyat menceritakan asal mula mengapa burung kakatua memakan hasil panen pada sebelum manusia sempat memakannya.
Jadi memang dia berasal dari manusia, anak kecil. Lalu pada saat dia naik di atas bubungan itu, setelah menyampaikan kepada orang tuanya bahwa besok lusa kalau orang tua punya tanaman, orang tua belum ambil atau manusia belum panen, saya harus makan lebih dahulu. Itu sayapnya sudah ada, langsung dia terbang sendiri.
Burung kakatua hinggap di jendela, nenek sudah tua, giginya tinggal dua.
Burung kakatua pun dianggap sebagai hama yang mengganggu kebun masyarakat. Dari beberapa cerita lainnya juga dikatakan bahwa kakatua pintar berbicara karena dulunya adalah manusia. Selain itu, manusia juga dilarang makan burung karena dulunya burung adalah manusia.
Seperti halnya pesan-pesan nenek moyang yang ada di sini, ketika kita berbincang-bincang dengan masyarakat tentang sejarah dan asal-usul dongeng yang ada di masyarakat sendiri, sedangkan nenek moyang sudah memesankan, memberi pesan konservasi terhadap kakatua govin ini sendiri, yaitu bahwa anak keturunan itu jangan sampai membunuh atau makan dari burung kakak tua Goviniana. Sehingga ini konsep sederhana yang diajarkan nenek moyang masyarakat di Tanimbar terhadap konservasi kakak tua Goviniana yang merupakan endemik Tanimbar.
Banyak tantangan untuk dihadapi untuk melindungi Kepulauan Tanimbar, tanah kakak tua Tanimbar. Kakak tua Tanimbar merupakan fauna endemik yang harus menjadi perhatian berbagai pihak untuk tetap melestarikan
Selamat siang Bapak Ibu.
Cek sound.
Siang, Pak.
Selamat siang Pak Cahyo.
Selamat siang.
Selamat siang, Pak.
Selamat siang.
Selamat siang semuanya.
Siang.
Siang, Bapak.
Selamat siang, Assalamualaikum warahmatullahi wabarakatuh. Welcome to Indonesia
Waalaikumsalam.
7 International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2020. In this session, we will have about 12 presenters and divided into 2 sessions. And before we start the first session, I will remind of all the participants to activate the video. Please activate your video so all participants can see our, can recognize our face, and please mute the microphone. And before the start for the question and answer, we can use chat, or I will read the question in chat after all the video presented by the presenters finish. So for the first presenter, we will have Bapak Mukhlisi with the assessment of earth and mammal in Wanaput Patra Lestari PT Pertamina Refinery Unit 5 Balikpapan. And Mukhlisi has already prepared the video for Mas Tulus or committee. Please prepare the video for Mas Mukhlisi. No voice.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, all participants of the virtual conference at the Indonesian Science Expo in November 18, 2020. My thanks and appreciation for moderator and all organizing committee in LIPI. At this moment, I will present a presentation entitled An Assessment of Bird and Mammal in Wanapatra Lestari, PT Pertamina Refinery Unit 5, Balikpapan. Introduction. Wanapatra Lestari is a forest fragment of 45 hectares in the working area of PT Pertamina Refinery Unit 5, Balikpapan. This forest is very unique and strategic, located between industrial area in the center of Balikpapan city. This forest also has a high historical value because in it there is the first location of oil well in Balikpapan that established in 19th century. Although not as a protected forest, Wanapatra Lestari has a high value of environmental services for the Balikpapan city, such as oxygen provider, carbon sinks, and also habitat for bird and mammal. The presence of birds and mammals in the forest around industrial areas can be an indicator of good environmental quality. On the other hand, these animals need to be periodically monitored as a basis for the better future environmental management effort. Thus, the study aims to analyze the diversity of bird and mammal species in Wanapatra Lestari, PT Pertamina Refinery Unit 5, Balikpapan. Method. Wanapatra Lestari is located among the oil refinery industry areas which are very important in Balikpapan. Geographically, the research site is in the front of Balikpapan Bay and directly faces the Makassar Strait with contour that tend to be hilly. The research area is characterized as a karanas and coastal forest. Data collection has been carried out on December 2017. Procedure. In this study, we use a combination of point count and VisNet for bird survey. Bird watching is carried out in the morning and in the afternoon. To complete the point count method for VisNet were placed purposefully around the research site. And then for mammal survey, data collection was conducted using live traps, camera traps, and also line transect method. A total of 25 small mammal traps are installed with the distance between traps about 30 meters. Meanwhile, as many as 10 camera traps were installed for 1 month. Data analysis. Bird and mammal data were analyzed by grouping into family, species, and conservation status. For bird data, each species was calculated for the value of relative abundance and relative frequency. We also analyzed feeding yield for each bird species identified. The conservation status was adjusted to national regulation under Permen LHK number P106, IUCN Red List, and also CITES. Result and discussion. For bird, we have recorded as many as 17 species of bird. Meanwhile, previously study report that in a research area only found 7 species of bird. The study at 14 species of bird that have not been recorded in previous study. So if we recapitulate with previous study, the composition of bird species in the research site were 21 species. However, in this study, there were 4 species of bird that were absent or not found from the list of bird species that had been recorded in previous studies. In this table, we can see if Pycnonotus aurigaster or cucak kutilang has the highest value both on relative abundance and relative frequency in the study location. Based on the grouping of the feeding gill, insectivore and also insectivore frugivore become the most common feeding gill, were found with a percentage of 29%. In the study, we only found Haliastur indus or elang bondol was categorized as a protected bird species and indexed in CITES Appendix 2. According to the IUCN Red List, all birds identified are still in the least concern category. For the result of the mammal survey, we identified a total of 6 mammal species. The majority of them are small mammal group, and they are species commonly found in various places, especially secondary forests and shrubs. If these findings are then added to the result of previous study, there are a total of 8 species. So this study also add new record to the mammal species to previous database, that is Cynopterus brachiotis or codocrawar, and also Tupaia minor or tupai kecil. And then based on conservation status, of all the mammal record in this study, none were categorized as protected wildlife species and CITES appendix. According to IUCN Red List, only Macaca fascicularis or monyet ekor panjang is in the vulnerable category. Meanwhile, other species are in the least concern category. Here are some pictures of the mammals in the study. There are musang pandan, bacing kelapa, codocrawar, and also monyet ekor panjang. Conclusion: in general, we found that the diversity of bird and mammal in the Wandapatra study forest has increased since the first survey conducted in 2015. For bird species, there have been an increase in baseline data of 14 species, while mammal are 2 species. To get more complete picture of biodiversity, monitoring activities still need to be done periodically in the future and also by involving other fauna. That's all. Thank you and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mas Mukhlisi, for the great presentation and great result for the bird study in Balikpapan. For question for Mas Mukhlisi will be after all the presenters present their results. So the next presentation will be presented by Yeni Aryati Mulyani, bird species used in songbird contest in Jabodetabek region, Indonesia. Please.
Good afternoon, ladies and gentlemen. My name is Yeni Amulyani. I would like to present our study entitled Bird Species Contested at Songbird Competition in Jabodetabek region. My collaborators are Tia Angguni, who unfortunately cannot attend the conference today, and Ani Mardia Stuti, who is now presenting in another room of this conference. And we all from Department of Forest Resources Conservation and Ecotourism, Faculty of Forestry and Environment. The background of our study is the fact that birds are popular as pets in Indonesia. A study by Jepson and Laddell showed that many inhabitants in big cities in Indonesia kept birds as pets. Compared to other animals, birds ranked highest. The fact is also supported by culture, especially in Java, where it is believed that an established gentleman should have pet birds or keep birds in a cage. Birds are kept as pets because of their interesting characteristics, mostly their song performance. The ability of birds to produce beautiful songs also made them valuable for song contests. Assessment in song contests include song rhythm, variation, song volume, song length, and physical appearance. Nowadays, song contest is increasing in Indonesia. It is shown by the more and the more events are conducted in big cities and even in suburban areas. There are several organizations that routinely or regularly schedule bird song contests, and this is one of example of brochure showing contest classes and the prize for the winner. The increase in bird contests is believed to have positive and negative impact. The positive impact are the improved knowledge of people on birds. People become more familiar with species, with races, bird bioecology, and especially breeding and maintenance. While the negative impact is the increased risk for survival or sustainability of birds in the wild through increase in illegal trade and poaching. Unfortunately, there are still limited studies on bird contests, such as what species are preferred, conservation status, etc. Therefore, our objectives are to identify bird species contested in Jabodetabek area and to classify them based on their conservation status. We chose the location in Jabodetabek region, which include Jakarta, Bogor, Depok, Tangerang, and Bekasi. And we explore information from social media to find out the schedule of bird contest event and also the organizing committee. And we contacted the organizing committee, and then we did observation during March to May 2018. And we visited 20 songbird contests. And we did some interview with the organizing committee and the participants. We analyzed bird species that we found, and we classified them based on origin and conservation status. The result shows that there are 15 bird species of 12 families used in the contest. 13 species were native and 3 were exotic. The 3 exotic were canary, lovebirds, and huamei. And the most frequent species to be contested is lovebird. We found lovebird in every contest that conducted during the period. In addition to birds contested, we also found one species, which is oriental dollar bird. And it is not for contest, but for sale in the contest area, because it is believed that this species is good for master species to train other songbirds. Among local or native species, greater green leaf birds are the most frequent used for contested, followed by white ramshama. Based on conservation status, we found 1 threatened species, which is greater green leaf bird, and 2 near threatened species, lovebird and chestnut-capped thrush. 1 species, greater green leaf bird, fall into protected species. All native birds are non-appendicitis, while huamei was listed in Appendix 2. Based on the interview, we found that the committee and also the participants have done some conservation-related efforts. For example, the organizing committee, they conduct ringed or marked class, and only birds from captive breeding can enter the competition. Birds produced from captive breeding will get a certification in the form of ring, or ban that apply to birds like, and it is applied when the birds was about 1 week old. So to make sure that it is from the captive breeding facilities. There are participants who also do small-scale captive breeding, especially for lovebirds, because lovebirds is relatively simple and not very high in cost. The conclusion of our study shows that the most popular bird species to be contested is lovebird. However, in general, local or native species are still preferred. Captive breeding efforts need to be encouraged, especially since we found protected species. There were no specific regulation on bird contest. Therefore, it is necessary to ask standardization of this activity, including regulation on species to be contested, origins, conservation efforts, and other criteria following animal welfare. Thank you.
Thank you, Ibu Yeni, for the presentation about the songbirds in Jabodetabek, and it's very common in Jabodetabek for songbirds competition. And the next presentation will be presented by Ibu Tika Dewi Atika, entitled Population, Economic Value, and the Trade of Dalbergia latifolia in West Nusa Tenggara, Indonesia.
Thank you, moderator. Good day, everyone. I would like to present our study on population, economic value, and trading system of Dalbergia latifolia in Lombok, West Nusa Tenggara, Indonesia. Introduction. Dalbergia latifolia, or rosewood, is high-quality timber. In Indonesia, we call it sonokling. It has a high price and recognized internationally as one of the world's most valuable timbers, used for high-end furniture, carving, and also music instruments. This species distributed mainly at Java and West Nusa Tenggara. Some literature reported that this species also distributed at East Nusa Tenggara, Sulawesi, and also Sumatra. Since 2017, this species was listed in Appendix 2 CITES, which gave a consequence on the management system. So our question here is, how is the population and the trade system this species after listed in Appendix 2 CITES? So our study aimed to provide information on population, economic value, distribution, and the trade of sonokling in West Nusa Tenggara. The material method, this study took in Lombok, West Nusa Tenggara, since 31st of July until 6 August 2018. In this area, we took sample from several sites here in Seoul, WL1 and WL2 are planted at agroforestry system, and it's managed by local people. In EL here, they planted in monoculture system like this, and it's managed by private company. In here, in WL3, they planted in mixed planted system, and it's managed by Forestry Regional Office or BKSDA, West Nusa Tenggara in protected area, Nature Park Bangko-Bangko. And we set up plots 2 until 5, depend on the situation, with the size 20 by 20. We measure and also record all sonokling trees which has girth more than 50 centimeter, and also we interview some traders, farmers, and also forestry officer. Result and discussion. We found that snorkeling in West Nusa Tenggara, mostly in hilly area at low altitude with rocky and dry soil. It was planted since 1970 and 1980s in reforestation programs. In WL3, snorkeling was planted in 1997 under the reforestation programs by the Forestry Office of West Nusa Tenggara Provincial Government. So this is the comparison of the good quality, which contain very big hardwood, while this is the tree with no hardwood. This is the table about the density of Dalbergia latifolia in each sampling site. The density is vary between 12 until 34 individual per 40 meters square. Here we can see that in AL has the highest density compared to others. Probably this is related with the management after planting. They did such as weeding, fertilizer, and so on. And here, for the seedling, we only found in sampling site WL3. So in this location, probably this is related with the forest condition. In this location, is very moist, so the seedling can survive even during the drought. This figure shows the size distribution in its sampling site. We can see here that the trees managed by local people, local farmer has small sampling, has small diameter and really large compared to others. Probably this is related with the maintenance after planting. In local farmer, they often cut the shoot for cattle, especially during the dry season. This table is about the wood categories found in West Nusa Tenggara. They sell the short timer wood with different forms. From square, board, and balok kaki. We can see here that the board form show the highest price. It can reach up until 50 million per cubic. Usually they sell board in a good quality, so which is characterized with a big size of the hardwood, while the others, such as in square or balok kaki, here they show a smaller size of hardwood. After this form, they will send it to primary or secondary industries in Java. So this is the trading system in West Nusa Tenggara on snorkeling. So before listed in Appendix 2 CITES, the farmer directly sell the wood to local buyer. And actually, previously the species is rarely traded, and usually they use the wood for firewood or furniture or something like that. And they just transport it directly to local buyer with the low price, about 2 million per truck. While after listed in Appendix 2 CITES, there are 4 trading system here. The striking difference is the presence of middlemen here, and for some of the farmer inform us that currently the middleman, this trade system is common in West Nusa Tenggara. And the traders also will set the price, usually until 10 million to 50 million per cubic. And up to July 2020, there are 22% and one industrial forestry plantation company on Rosewood categorized as the trader register in West Nusa Tenggara. Since this species was listed in Appendix to CITES, the transport should follow the decree of Minister of Forestry, with number 447/2003. And also here, the provincial government of West Nusa Tenggara has published a new regulation through notification letter to ensure the traceability, legality, anticipation of illegal logging or poaching of rosewood and deforestation, which establish the Pemberantasan Perusakan Hutan or P3H Task Force, with assigned to verify and clarify the legality of the wood. The P3 Task Force members are from BKPH, Kodim, Polres, and BKSDA. So so our studies conclusion are that Berdaulat latifolia in West Nusa Tenggara was planted with the density ranging from 12 until 34 individual per 4 meters square. There has been an increase in trade price since listed in Appendix 2 CITES. There are 4 trading system of snorkeling at West Nusa Tenggara. The last is, since listed in Appendix 2 CITES, the provincial governor West Nusa Tenggara has issued the notification letter to ensure the traceability, the legality of the source by establish the P3H Task Force. Thank you very much.
Thank you very much, Ibu Tika, for the presentation regarding the snorkeling, especially after the listed in Appendix 2 CITES and change the trading system. It's very interesting topic, and it's hopefully will be used for the management of snorkeling in the future. So in the next presentation, we will have Ibu Titin Ngatinem Partoswiryo, entitled Botanical Inventory and Rarity of the Fern genus Pteris in the Karst Forest of Bantimurung, Bulu Sarawung National Park, Indonesia.
Bismillahirrahmanirrahim. Ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. I pleased to talk about botanical inventory and rarity of the fern genus Pteris in the Karst Forest of Bantimurung, Bulu Sarawung National Park, Sulawesi, Indonesia. Sulawesi, formerly known as Celebes, is one of the most poorly known ecoregions, but has been highlighted as globally important biodiversity hotspot and conservation area. Plant species collection rate on the island are among the lowest in Indonesia. There are about 23 specimens per 100 square kilometer. The pteridophyte flora of Sulawesi is fully documented. The exact number of pteridophyte Sulawesi is still uncertain. Therefore, Cato estimates that Sulawesi is home of about 500 pteridophyte species. Therefore, the forensic studies for clarifying the diversity of ferns and fern areas in Sulawesi are still needed. Pteris is large genus as it is consisting of about 250 species. It is a cosmopolitan genus and distributed throughout the tropical, subtropical, and temperate areas of all continents except Antarctica, from Australia, New Zealand, South Africa, northward to Japan, and North America. Pteris grow either terrestrially or lithopitically on rock on a diversity ecosystem. The genus is characterized by its marginal story, supported by collecting vein, and protected by inoncium formed by the reflected lamina margin. Pteris is interesting genus as it is known to be model for studying population, for studying plant evolution and speciation. Species diversity of Pteris has resulted in part from hybridization, apogamy, and polyploidy process. Why should we concern with these rare species? Understanding the ecology of rare species can inform aspects of conservation strategies as well as population status. Forensic study on fern genus Pteris in the karst forest of Pantimurung Pulau Sarong National Park was carried out to aid our knowledge of ferns flora of Sulawesi. This study aimed to conduct an inventory of Pteris growing in the karst forest of Pantimurung Pulau Sarong National Park and determine the rarity of each species. Pantimurung Pulau Sarong National Park is a national park located in Maros and Pangkep Regency, South Sulawesi Province, about 50 km north of Makassar. Random search with purpose sampling was applied in observing the rare species of Pteris. Specimens further were deposited at PO on the Hipparium of Bogor Garden, POHB, and plants were identified by referring from flora of Malaya, Ptidaraceae of China, and also referring the specimen of Pteris deposited in Hipparium bogorense. Research and discussion. Zhang et al. Classified Pteris into three sections, namely section Campteria, section Pteris, and section Quadriauricula. The three sections are mainly differentiated by typical of veins, segment marking characteristic, the extent of the spine of the costa, and the injection of spinae or pinule. Pteris section Pteris are characterized by morphological variation as follows: venation free, pinnae or pinule anterior with cartilaginous margin. These are Pteris pellucida and Pteris radicans. Pteris section Quadriauricula are, aduh, ini salah lagi. Oh, pasti. Pteris section Quadriauricula are determined by using morphological characteristics as follows: pinnae and pinule pinnatically divided or lobed on at least one side. Segment lanceolate, often obtuse or acute, without cartilaginous margin, anterior or rarely serrated. The third section is section Campteria that can be recognized by the character combination as follows: costa spini along both sides of group On adaxial side, venation anastomosing to form a series of narrow areolas along costa, vented free beyond areolas and extending to margin. Table 3 showing Pteridacea of Karst Forest of Resort Biology, Pantimurung, Pulau Sarong National Park. Two species that are relatively widely distributed are Pteris informis and Pteris vittata. Pteris longipes, Pteris lucida, Pteris huatri aurita, Pteris radican, Pteris spinascan are considered as rare species. Conclusion: 9 species of Pteris were recorded from 10 sites of Pantimurung, Pulau Sarong National Park. Pteris longipes may be new record for Sulawesi. Further studies are needed. Pteris longipes, Pteris pellucida, Pteris huatri aurita, Pteris radican, and Pteris spinascan are considered as rare species. This is all my presentation. Thanks for your attention and listening.
Thank you, Bu Titin, for the presentation about the ferns in Karst Forest in Pantimurung, Pulau Sarong National Park. And we will have the next presentation will be Ibu Yelin Adalina, entitled on analysis of vegetation and land use under Pinus mercusii stand at KPH Malang.
Hello everyone, my name is Yelin Adalina from Forest Research and Development Center. On this time, I would like to presentation about analysis of vegetation and land use under Pinus mercusii stand at KPH Malang. On this research, I am the main author and Reni Sawitri is the co-author. Introduction. Forest management must provide economic, ecological, and social benefit for the community surrounding the forest. The problem of community surrounding forest was a lack of land. Pine mercusii is one of the mainstay tree species distributed by Perum Perhutani as a state-owned enterprise in the forestry sector. Perhutani in Kesatuan Pengelolaan Hutan Malang empower communities surrounding around the forest to do agroforestry system at Pine Stand through community-based forest research management program. The community forest management system impact increasing the income of farmers around production forests. The study aimed to find out the structure and composition of the pine stand plantation managed by Perum Perhutani in KPH Malang and to know the pattern of land use under the stand of pine mercusii by communities around the forest. The study conducted in Sumbergondo village, administratively located in the Junggo Forest Management Resort, Singosari Forest Management Unit, Panjat Forest Section, Forest Management Unit Malang. The methods are: first is vegetation analysis. Vegetation analysis conducted in plot 53, 54, and 56. Vegetation analysis was performed at each level of tree growth with classification, seedling, sapling, pole, and trees. The collected data was analyzed to determine individual density, species frequency, and dominance. Second is socioeconomic. Interview conducted of using questionnaire or in-depth interview. The number of respondents interview was 30 respondents. The research village was chosen purposefully based on the use of the land under pine mercusii. Land use carried out through agroforestry pattern. with type of agricultural plant, fruit, and herbs. In addition to the respondent, in-depth interview conducted with other stakeholders involved in forest area management, including community leaders, the staff of Perum Perhutani, and village head. Third is data analysis. First is important value index for pole entry and important value index for seeding and sampling based on the formula. Result and discussion. The table show about vegetation analysis. Pine forests in KPH Malang have density for tree level 214 individual per hectare, for level 63 individual per hectare, and sampling level 35 individual per hectare. Pine stand in agroforestry had average diameter of 29.7, average tree head of 12.6 meter. There were 5 species of plant at the pole level, consisting of avocado, coffee, pine, silk tree, and suren. This shows that the species of plant have adaptability in the forest area. Based on the analysis of seedling vegetation, 45 species of undergrowth obtained of FMU Malang, consisting of 15 types of plant cultivated by the community. The sampling level, the vegetation consists of avocado, coffee, banana, pine, and mango. The highest important value index analysis result at the state level are coffee plant, with a density of 18 individual per hectare. The sampling level show that the coffee plant has high adaptability than other species. Society have long used the area as intensive agricultural land for vegetables, various types of flowers, banana, corn, coffee, and ginger. A total of 359 farmers are working on the ground in the area managed by Perhutani Public Corporation. The study result that as many as respondents planting one type of cultivated plant is 56% and 43% respondent plant 3 types. The highest income obtained if the respondent plant 2 species of plant is broccoli and carrots. The amount of respondent income from farming depends on the number of plant cultivated, type of plant, maintenance, and fertilization of plant. Data show that broccoli has the highest selling value compared to other types of plant. The income from farming with an intercropping system under pine stand contributed 12 until 46% to the respondent total household income with an average of 34%. The data show about socioeconomic of communities. All respondents are Javanese and Muslim. All respondents don't own arable land that is outside the forest area. The average income of respondents was 1,600,000 per month. The activity of agroforestry system by cultivated land in the pine forest provided an additional income for the community of 1 million until 6 million. So it helped increase income promising prosperity. The conclusion. Characteristics of individual respondents are relatively homogeneous. The average respondent income is below the regional minimum wage of Malang City. The result of the analysis of vegetation include 5 types of plant at the tree level, 5 types of pole level, 5 types of sampling level, and 13 species of undergrowth. Pine dominate the highest important value index. Pine Mercus, which is a staple plant in forest vegetation. Pine trees become the main stand that plays a role in providing shade to the cultivated plant below. Most people cultivate land in the forest area under Pine Mercus stand. with intercropping pattern consisting of various types of agricultural plant, ornamental plant, and fruit plant. Respondent income from farming under pine stand is 1 million until 9 million from arable land. Land use under pine stand with the intercropping system is an optional land use system and impact the economic of the community around the forest. Thank you very much.
Thank you, Ibu Yelin, for the presentation, and will remind all of us, if you have any question for all presenters, please drop in the chat, or maybe you can use raise hand if we still have a time for the discussion in the after the last presenters. And the last presenters will be presented by Ibu Rinda Kirana, entitled Genetic Purity and Population Structure of Potato Variety Granola L.
Good afternoon. I would share our research with the title Genetic Purity and Population Structure of Potato Variety Granola L. I am Rinda Kirana from Indonesian Vegetable Research Institute, and the other authors are Kusmana, Asih Kartasi Karyadi, Nikagdi Gunadi, Refli Nur, and Catur Hermanto. This is Granola L cultivation. This variety released 1993 with high yield around 26 tons per hectare, resistant to viruses and widely grown by Indonesian farmers. The problem is, as a variety that has been released for decades, some farmers reported that the performance of Granola L variety has been phenotypically changed. Kusmana et al. Found the phenotypic change of Granola L in several characters, such as sprout, leaflet, flower, and also tuber. This is the example, the phenotypic change in sprout character. The research question is, how about genotypic? The Indonesia Center for Agriculture and Genome, known as PGPI, has successfully performed whole genome sequence of 5 Indonesian potato varieties. Based on the genome sequence, a set of microsatellite markers distributed on 24 potato chromosomes has also been developed. And the objective of our research are to assess the genetic purity and determine the population structure of Granola L variety using microsatellite marker. Our method were planting 12 potato genotypes from 12 different sources, DNA isolation using CTAP method, PCR analysis using 18 SSR with optimum PCR program, fragment analysis, reading molecular weight by gene analyzer program, and assess genetic purity and also determine population structure. This is our research, planting the material conducted in Lembang, and this is a DNA isolation. After that, we running PCR, and product PCR, we conducting the fragment analysis. For the genetic purity, this is the dendrogram that show genetic purity with similarity coefficient value 0.98 reflect genetic variation value of 0.4. These results show the low genetic variability or high genetic purity among 12 granola L. For population structure, this is the result from structure program. The two subpopulations obtained in the structure analysis reflect 12 granola L have narrow population structure and low genetic diversity in the population. There are two reasons to explain our result. Granola still be favored by farmer, although phenotypically different. Breeding program using granola L as a parent. Usually, interrogation of one or two disease character for create the new varieties. The continuous manipulation activities in one character can cause narrower genetic basis. And the second, the low number of SSR and sample used on this study might be contribute to the high genetic similarity. The result of this study indicate that the granola L variety still has high genetic purity and narrow population structure. For the next study recommended include more SSR and more sample. We are very much thankful for IFEGRI to financial support and laboratory technical support by Dinda Mutiara Sakti and Nira Nuraini and data analysis support by Pepen Effendi. Thank you for your attention.
Thank you very much, Ibu Rinda, for the presentation. And after we have 6 presenters here, we still have about 15 minutes for question and answer. And I see in the chat room we have one question. And if all of you have a question, you can raise your hand or just write the question in the chat, and I will read and maybe directly for the presenter to answer the question. And the first question from Ibu Yeni to Pak Mukhlisi. The question was, what factor that might influence the change in diversity compared to previous study? And the second question is, did you do spirit analysis in calculating the abundance of birds using point count and misnetting? If so, which method that give the better results? Maybe Pak Mukhlisi can answer directly in the question from Ibu Yeni. Please open your mic.
Yes, thank you. The question from Ibu Yeni Aryati Mulyani. The first question, what factor that might influence the change in diversity compared to previous study? I think the influence factor change diversity generally is the increase in human activity around the research sites. Currently, Pertamina is expanding the refinery oil so that there are many human activities. In addition, it may be due to different in sampling method and size sampling. And the second question, in this study, we found that point count give a higher increase in species abundant than misnet, but some elusive birds are hard to observe. So it's easier to observe by misnet. And I think a combination of the two methods is best to observe the bird.
Any comment from Ibu Yeni, please, if would like to elaborate the answer from Pak Mulisi, or if anyone have a question, please feel free.
Thank you, Pak Cahyo. Regarding the changes in diversity, is there any specific reason why some species are still there, or some species cannot be found anymore? Are those only because of that human, or is there any management effort from the Pertamina itself? Thank you.
Okay, please, Pak Mukhlisi, if with a brief and concise answer.
Unfortunately, in the study, we don't correlate about species abundant and environment, so we can observe the correlation between recent absence or bird and environment condition.
So no management effort from the Pertamina to maybe to increase or to improve the habitat?
Yes, I think the management effort also increase the species abundance. They are making the ecotourism in the forest area, so they have protected the area by some employment dari karyawan.
Okay.
Thank you.
Thank you, Bu Yeni dan Pak Mukhlisi, for your question and answers. And there is one participant raise hand. Ibu Tika, please direct question.
Okay.
Open your mic and please brief and concise.
Okay, Pak Mukhlis, there is one question. Did you research the food viability for the related to the bird diversity?
Okay.
So did you do research about the food viability, such as what kind of plant that you planted? Probably it related like that. Thank you, Pak Cahyo.
Okay, thank you, Bu Tika. Pak Mukhlisi, please. Your so favorite today.
In the study, we just observe the feeding gill, the bird, but we can observe the food availability. But we see some plant visit some birds, especially casuarina, it seems like cemara laut, and makaranga or mahang.
Okay, that's it.
Thank you.
Pak Mukhlisi. Okay, Bu Tika, thank you very much for the question, and thank you, Pak Mukhlisi, for the answers. And there is one question from chat from Ibu Diah Wulandari from Biotechnology Research Center of LIPI to Ibu Rinda. In your opinion, if the genetic purity in granola potatoes is still high, so what factor cause the morphological change that occur in the land? Ibu Rinda, please answer directly. Open the mics.
Okay, thank you for the question, Ibu Diah. In our study, yes, the different occur in character, morphological character, and I think because of the environment, and also in this study we use different seed sorts, 12 different seed sorts, and maybe the management of seed sorts on the previous before the study can affect the morphological change. Okay, that's it.
Okay, thank you, Bu Rinda. Bu Tia, if you have any more comments or want to elaborate the answer from Ibu Rinda, please open your mic directly.
Thank you. It is clear. It is clear enough. Thank you.
Okay, thank you, Bu Rinda and Bu Tia for the question and answer. And still have a time. Again, Bu Tika, want to have a question? Bu Tika.
Okay, thank you, Pak Cahyo. I would like ask to Ibu Yenli. So in your slide, you mentioned that the income of the farmer is below. Is it related with the yield production? Since we know that pinus will release a secondary metabolite, which will give an effect to the growth of the crop. Yeah, is that like that?
Okay, thank you, Bu Tika. The question for Ibu Yelin regarding the vegetation and use on Pinus mercusii. Please, Ibu Yelin. Ibu Yelin, still in the room? Ibu Yelin? No? So, Ibu... Okay, Bu Yelin, please.
Would you like, please say again about the
So
question?
my question is it related? Is it there is a related between the low income of the farmer with the yield production?
Oh, I see.
Yes, since we know that Venus mercusii will release secondary metabolite secondary, like that. Thank you.
Yes, it's related about the income with many kinds of species. This is if the intercropping is only one species, it's a low, and if the intercropping 2 or 3 species, it's the high income. But that I thought they have like that because they still what's this, the investment of there is related to. Okay, thank you.
Thank you, Bu Yelin. Ibu Tika, still one more elaborates the answers or no? We still have 5 minutes from the schedule, so if there are...
Thank you.
Okay. Okay, thank you, Bu Tika. We still have 5 minutes for question and answer before the next session for the next 6 presenters. If we have no more question, maybe I would like to... Okay, one more question from Pak Mukhlisi from Baliteka SDA to Ibu Yeni. So it's just like a TikTok between Pak Mukhlisi and Ibu Yeni. Does the songbird that is contested, especially leaf bird, come from Java or another island? So it's interesting questions because songbird usually come from many islands. So Ibu Yeni, please.
Okay. Thank you, Pak Mukhlisi. Thank you, Pak Cahyo. Actually, in this, in our research, we didn't trace back where the bird came from, but we interviewed the owner, and they just say that mostly like 80% of the bird that they used to for contest, they got from they bought it, and only there are like 1% they captured in the wild, and the wild one they captured here in Java. So we didn't really know whether that leaf birds is originated from Java or Sumatra or Kalimantan. We could not trace the origin of the contested birds. I think that's what I can inform.
Okay, thank you, Bu Yeni. I think the answer is already clear enough to about the origin of the songbirds. So last question from Pak Sutomo from Kebun Raya Bali to Ibu Tika. Are there other use of Dalbergia in terms of its ethnobotany aspect in your study sites? So it's like about the ethnobotany and relating to snorkeling. Please, Bu Tika.
Okay, thank you, Pak Tomo. In West Nusa Tenggara, we only discover one use for Dalbergia is for firewood. Only for that. Firewood, food kettle, and furniture. Only that. Thank you, Pak Cahyo. Probably on.
Okay. I think Pak Thomas still more question. We still have 2 minutes if you want to elaborate the answer from Ibu Tika.
No, thank you, Mr. Cahyo. Thank you.
Okay. Thank you very much for all presenters and all for participants in this room. I think we have already finished in session 1 for this session, and we will move forward to the next session. We will have 6 presenters. We will have from time from 3 to 20. So the first presenters will be Bapak Ruby Herianto, and title of Genetic Diversity and Relationship of 12 White Turmerics, Curcuma sedoaria, accessions based on morphological traits.
Good afternoon, ladies and gentlemen, participants of the international conference. Let me introduce myself and my partner. I am Ruby Herianto, and my partner is Siti Fatimah Sahid. We are from Indonesian Spice and Medicinal Crop Research Institute. In this opportunity, I would like to present my research entitled Genetic Diversity and Relationship of 12 White Turmeric, Curcuma sedoaria, accession based on morphological trait. This is my outline. First, I would like to talk about introduction, second, material and method, third, result, and finally, conclusion. Introduction. White turmeric is one of rhizomatous herbaceous perennial plant of the ginger family, Gingiberraceae. Nowadays, the plant widely cultivated in Asia, especially in South Asia and Southeast Asia. White turmeric is a large plant having a height of 70 until 100 cm, pseudostem of 30 until 35 cm, and green seed. Propagation of this plant using rhizome. White turmeric can be useful as a spice and medicine. As a spice, white turmeric in Thailand for curry pasta, while in India for curry dye and flavor additive in seafood dish or Indian cuisine. As a medicinal, especially traditional medicine, white turmeric for improvement of blood circulation, menstrual flow, abdominal cramps, and rheumatic pain. The useful part of white turmeric is a rhizome. The chemical compound of white turmeric is essential oil, 1 until 1.5%, curcumin, gum, resin, starch, and tannin. The curcumin in the white turmeric rhizome has many functions such as anti-cancer, anti-inflammatory, antihypertensive, anti-diabetic, antimicrobial, immunodilator, antifungal, anti-proliferation, and nephroprotective. Germplasm is an important component in plant breeding programs, where assembly a superior variety requires a wide genetic diversity. Genetic diversity can be measured in several ways. However, the main source for genetic diversity is strong morphological character. In addition, information from genetic diversity will also be very useful in conservation strategies and future breeding, because conservation and breeding should be done after knowing the genetic diversity. This study aimed to investigate 12 accessions of white turmeric has a genetic diversity and relationship among their plant. Material method. The material used in this study is 12 white turmeric accessions collected from 7 different areas in Indonesia. This experiment was conducted from 2014 until 2015 planting season on the Curug Research Station of Indonesian Spice and Medicinal Crops Research Institute. The experiment was arranged in a randomized block design with 3 replications. Observation of morphological trait. The study was conducted with a descriptive method which observed the morphological character of plant. The parameter divide 2 groups. First, parameter of growth plant are plant height, leaf length, leaf width, number of leaves, leaf thickness, stem diameter, and number of tiller. Furthermore, the parameter of yield component is represented in rhizome weight, rhizome length, rhizome width, and rhizome thickness. Statistical analysis, the measurement of genetic diversity, correlation coefficient, and cluster analysis using R software. Result. Table 1. Show the morphological variation of 12 white turmeric accessions. We can see parameter of rhizome weight is the highest of coefficient of variation with value 30.15%. Parameter number of tiller have a coefficient of variation with value 25.16%. This value is not high. This indicates that genetic diversity occurs in rhizome weight and number of tiller because the value more than 25%. Table 2. Describe genotype, phenotype, and environmental variance and coefficient of variation, broad sense heritability of 12 white turmeric accessions. The diversity of value based on the value of the genotype, coefficient of variation, and the phenotype coefficient of variation. Genotype and phenotype diversity of 12 white turmeric accessions is allowed that shown the value of the genotype coefficient of variation and phenotype coefficient of variation for all observations under 25%. Low heritability with a value under 0.25 indicated by leaf thickness, rhizome width, and rhizome thickness. Meanwhile, other observations indicate the medium heritability, about 0.34 until 38. Table 3. Describe the correlation between traits. Rhizome weight trait has a significant correlation with stem diameter, rhizome length, rhizome width, and rhizome thickness. Other components correlate with the rhizome weight, but showed not significant. All morphological characters show a positive correlation. It is shown that the morphological traits of white turmeric interact with each other. Figure 1 shows the dendrogram of 12 white turmeric accessions based on morphology. 12 white turmeric accessions were clustered into 4 major groups. Is cluster 1, 1, 7, 8, 11, and 9. Cluster 2, 2, 3, 10, 12. Cluster 3, only 6. And cluster 4, 4, and 5. The biggest group in cluster 1 consists 5 members, and the smallest are cluster 3 consists 1 member. Figure 2, principal component analysis of 12 white turmeric accessions based on morphological trait. The result PCA divided 4 major groups, but the group is different with the dendrogram. We can see In dimension 1, we can see only 1, 9, and 7. Group 2, 2, 8, 11, and 3. Group 3, 4 and 6. Group 4, 10, 12, and 5. We can see youth component refer to accession number 1 and 7. Meanwhile, growth component refer to accession number 8 and 11. Conclusion. Region weight and number of tiller characters show genetic diversity with a coefficient of variation of more than 25%. 12 white turmeric accession divided into 4 groups. An achievement. The author thanks to Dr. Nuryani Bermawi, Endang Hadi PNT, Sri Wahyuni, and Wawan Lukman for the contribution in the exploration activities of white turmeric germplasm. This research was funded by the Indonesian Spice and Medicinal Group Research Institute, the Indonesian Agency for Agricultural Research and Development. Thank you for your attention.
Thank you very much, Pak Ruby Herianto, for the great presentation about the genetic diversity and relationship of 12 white turmerics. And the next presenters is Ibu Ruliana Susanti, entitled on preliminary study on plant ecology in Tangkahan area Mount Leuser National Park.
Good afternoon, ladies and gentlemen. My name is Juliana Susanti, and today I represent my other colleague who would like to present part of our research collaboration result between Kagoshima University and Indonesian Institute of Sciences. The title of my presentation is Preliminary Study on Plant Ecology in Tangkahan area Mount Leuser National Park. Indonesian biodiversity faces direct and indirect threat due to change of forest cover. Forest area in Sumatra, as one of large islands in Indonesia, also suffered the same threat. Previous study illustrate the forest cover changing in Sumatra area for the past 20 years, as we can see in this picture, from 1990s to 2010. If we see closer, the northern part of Sumatra has rather better condition compared the other part of the islands. This condition is due to the large number of conservation area and protected forests in the northern Sumatran region, including Gunung Leuser National Park. The national park was established in 1980 and covered for more than 10,000 kilometers square of forest. This is the biggest national park in the northern part of Sumatra. There are few studies on plant ecology in this area. Previous plant study in the area was conducted in the Katambe forest around here in around 1980 to 1990s. The other study that conducted in Gunung Leuser National Park mostly related with protected animals such as orang-utan and Sumatran tiger. So the purpose of the project or the purpose of this study is to understand the plant structure and composition in the tropical forest of Gunung Leuser National Park. Our study site was focused in the Tangkahan area over here in the eastern side of the national park near the border of the national park. If we can see here is the picture taken from the Google Earth. This area is the Tangkahan area. This is the visitor center, and this Bulu River, this river was named is Bulu River, is part of the border of the national park with the oil palm plantation just outside the national park. So if we can see here, this area is oil palm plantation, and this area, which is greener, is part of the national park. We established 0.5 hectare plot at the altitude of 320 20 meters above sea level and measure every tree with diameter over 4.8 centimeter. The identification was conducted in Herbarium Bogoriense, LIPI, and the data analysis was conducted using R. This is represent our research result. This table shows the total number of trees. This table shows the summary of our plot data. The total number of trees found in the plot is 690 trees or individuals, come from 175 species from 49 families. The total basal area of the plot is 42.90 meters square per hectare. The biggest and the tallest trees found in the plot is Sorea parvifolia from the Dipterocarpaceae family. Result on the counting of diversity index over here confirm that the forest has a relatively high diversity. The Shannon diversity index or H-accent more than 4 and the Simpson diversity index or D closer to 1 are rarely found, and it indicates strongly high diversity. The graph on the left shows the tree distribution according to the class diameter. We can find that 60% of trees found in the plot has diameter less than 10 cm, and 1% of the trees has diameter more than 80 cm. This result shows the dynamic of trees in the plot, and similar were with other research conducted in Ketambe area on the western side of the national park. This figure shows 10 families with the biggest basal area. The biggest basal area can be found in the plot is for Dipterocarpaceae family, cover 37% of the total basal area in the plot, followed with Myrtaceae and Achariaceae. This result is slightly different with the western part of the national park in the Ketambe area. To understand more about the forest structure, analysis on correlation between diameter and tree height were conducted, and the result shows strong correlation between diameter and tree height, with the coefficient correlation is for 0.82. Trees with diameter less than 50 meter mostly stand less than 30 meter high, and trees with diameter more than 80 centimeters stand more than 50 meter high. Some species can be considered as an emergent species. This is an example of emergent species, including Sorea multiflora, Irvingia malayana, Sorea parvifolia, Scapio macropodum, and Caralia brachiate. Next, this graph shows some families with the highest species richness and density. Again, Dipterocarpaceae was the most diverse trees family in terms of species richness and density also. Pilantaceae, Lauraceae, Ebenaceae, and Anacardiaceae have high species diversity, however, low in number of individuals. The Clusiaceae species, Achariaceae, Fabaceae, and Euphorbiaceae have large number of individuals but low in species diversity. Based on this research result, we can conclude that the forest area was dominated by Dipterocarpaceae species. In terms of diversity, it composed 30% of the whole population inside the plot. There are 16 Dipterocarpaceae species can be found in the plot, come from 3 major genus, namely Dipterocarpus, Sorea, and Fatica. picture shows Dipterocarpaceae species that can be found scattered in the plot. So we can see here the red dot represent Dipterocarpaceae and the blue dot represent the other species inside the plot. And these two pictures shows example of the Dipterocarpaceae family found inside the plot. This picture represent Fatica lowi, and this is the Sorea leprosula. It is very rare to found Dipterocarpaceae in the stage of flowering, so we are very lucky at that time. Thus, we can conclude that the forest area in Tangkahan has high tree diversity with a total of 175 tree species from 49 families. The plot was dominated by Dipterocarpaceae and show a dynamic forest condition with large number of small trees. We found emergent layer that was composed by Sorea porfifolia, Sorea multiflora, Irvinia malayana, Scapio macropodum, and Caralia brachiate. Finally, this is the end of my presentation. I would like to express my gratitude for all peoples and organizations that already help us in conducting this research. Thank you very much for your attention.
Thank you very much, Ibu Ruli, for the very great studies and also very well prepared presentation. I think the presentation is very friendly for all of us, very eyes friendly with color combination, etc. So hopefully the next presentation will have also very well prepared presentation. Ibu Siti Sundari, entitled Monitoring of Ecosystem Type in Tobalik Region, North Sumatra.
Good afternoon, everyone. Thank you very much. I am Siti Sundari from Research Center for Biology LIPI. According to the reviewer comments on our manuscript, we have to change our title. So our title is Monitoring of Structure and Composition of Mountain Forest in Tobalik Region, North Sumatra. This is the introduction. Terrestrial ecosystem around Tobalik is estimated to have a high level of biodiversity. It is also known that there are endemic biotypes and endangered species around the Tobalik area. The forest area around Toba Samosir Regency has a good forest character with standing stands. However, the forest area is still under pressure because of land development and management. This will directly have an impact on the existence of biodiversity, especially on the vegetation and the availability of water sources for Tobalik. The study aims to maintain biodiversity and obtain information about the condition of the forest around the Tobalik region. We conducted a monitoring study of permanent plots that had existed in 2018 in 3 villages of Toba Samosir Regency. This is the study sites. There were 3 villages. First village was Janggadolok. There were 5 plots in Janggadolok village. And the second village was Ambar Halim. There were 4 plots in Ambar Halim village, and the last is Halado village. There was only 1 plot in Halado, so we have 10 plots. And the method, first, vegetation remeasurement, 10 plots with a size of each plot was centimeter multiply centimeter. There were 9 subplots inside each plot with a size of 10 meter multiply 10 meter of each subplot. Parameters measures including the circumference of the trunk, the total height of the tree, and the height of the free branch. Second, data collection of new vegetation or recruitment and dead vegetation. Data collection on recruitment and dead vegetation were carried out in each subplot in the study sites. Recruitments were recorded by giving alumni number, collecting specimens, identifying the species, and recording the position of X and Y in the subplot. The total height of the tree and the free height of the branches of recruitments were also measured. Specimen taken were then processed in alcohol and sent to the Herbarium Bogoliense Research Center for Biology in Cibinong for further identification. The last is calculation of relative density, relative frequency, relative dominance, and importance value index or IV. Next is the results and discussion in plot 1, Jangkadolok Village. This is the result between 2018 and 2019. 3 species with the highest IV, and this is the density in 2018 and 2019. And this is the 3 species distribution in the plot 1 in Jangkadolok Village. And this is the number of recruitment and dead vegetation. And this is the result in plot 2, Jangkadolok Village, 3 species with the highest IV, and this is the density in 2018 and in 2019. This is 3 species distribution in the plot 2 in Jangkadolok Village, the number of recruitment and dead vegetation. And this is the plot 3 results in Jangkadolok Village, 3 species with the highest IV in 2018 and in 2019. And this is the density in 2018 and in 2019. And this is the 3 species distribution in the plot 3 in Jangkadolok Village, the recruitment and the dead vegetation. And this is in plot 4 in Jangkadolok Village, 3 species with the highest IV and the density result in 2018 and in 2019. And this is the 3 species distribution in the plot 4 in Jangkadolok Village. And this is the recruitment and the dead vegetation. And this is the last result in Jangkadolok Village in plot 5. And this is the 3 species with the highest IV in 2018 and in 2019. And this is the density result in 2018 and in 2019. And this is 3 species distribution in the plot 5 in Jangkadolok Village, and the number of recruitment and dead vegetation. And we move to the Ambar Halim Village, plot 1, the result of 3 species with the highest IV in 2018 and in 2019. This is the density result in 2018 and in 2019. And this is the 3 species distribution in the plot 1 in Ambar Halim Village. And this is the plot 2 result in Ambar Halim Village in 2018 and in 2019. Also for 3 species with the highest IV and the density. And this is the 3 species distribution in the plot 2 in Ambar Halim Village. This is the recruitment and dead vegetation. And this is the result in plot 3 in Ambar Halim Village in 2018 and in 2019 for the 3 species with the highest IV, and this is the density result in 2018 and in 2019. And this is 3 species distribution in the plot 3 in Ambar Halim Village, the number of recruitment and dead vegetation. And this is the result in the last plot in Ambar Halim Village, that was plot 4, and this is in 2018 and in 2019 for the 3 species with the highest IV, and this is the density result in 2018 and in 2019. And this is the 3 species distribution in the plot 4 in Ambar Halim Village, the number of recruitment and dead vegetation. And then this is the last plot in Halado Village, the 3 species with the highest IV in 2018 and in 2019. And then this is the density result in 2018 and in 2019. So this is the 3 species distribution in the plot of Halado Village in 2018 and in 2019 with the number of recruitment and dead vegetation. And this is the relationship between the number of individuals per hectare and diameter class per each plot. And the plots were dominated by vegetation with a diameter of 5 until less than 10 centimeter. All the plots are the same. Here, 3 species with the highest IV at the 3 study site in 2019 were mostly the same as the previous year in 2018. Difference in the density in 2018 and in 2019 and the number of individuals between recruitment and dead vegetation were small. Besides that, the plots were dominated by small trees. It means that forest dynamics have not seen significant changes due to the short time of monitoring schedule, that 1 year only. It is also probably because of anthropogenic factor that the forest is still well preserved by the community. And this is the conclusion. The plots in the study sites were dominated by vegetation with a diameter between 5 until less than 10 centimeter. The density and the number of recruitment and dead vegetation in Ambar Halim plot was the highest among the sites, followed by Jangpadolok and Halado. The overall stem composition and structure of the plots in the 2019 was mostly the same as the previous year in 2018. Okay, that's all our presentation today, and thank you very much for your attention.
Thank you very much, Ibu Siti Sundari, for the presentation. And then the next presenters is Ibu Suluh Nurmasiwi with the title Growth, Stomata, and Trichome Characteristic of Diploid and Tetraploid Artemisia annua.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name is Suluh Nurmasiwi from Research Center for Plant Conservation and Botanic Garden, Indonesian Institute of Sciences. I would like to present my activity with my colleague, Muhammad Effendi and Wiguna Rahman, also from Plant Conservation and Botanic Garden, Indonesian Institute of Sciences, Erwin Al-Hafiz and Trimuji Ermayanti from Research Center for Biotechnology, Indonesian Institute of Sciences, Raden Arthur Lelono from Research Center for Chemistry, Indonesian Institute of Sciences, and Nanang Yunarto from Center of Biomedical and Applied Health Technology, National Institute of Health Research and Development, Ministry of Health, Republic Indonesia. Our topic is growth, stomata, and trichome characteristic of diploid and tetraploid Artemisia annua plant. Artemisia annua is the only species of genus Artemisia to synthesize artemisinin. Since 2001, artemisinin has been recommended by World Health Organization to fight malaria in the country where highly resistant strains of the protozoan have been certified. The critical feature of the artemisinin as widely used anti-malaria compound is that artemisinin has shown no significant side effect. Artemisia annua is native to China and naturally grown in the subtropical and highland area, but nowadays it's successfully cultivated in the tropics such as Indonesia. Some cultivar of Artemisia annua were cultivated in Indonesia has low artemisinin content from 0.1 to 0.3%. It has become one of the problems of cultivation in Indonesia. Artemisinin is mainly extracted from the leaves which produce artemisinin in granular trichome. A breeding program to develop a plant that produces a high level of secondary metabolic produce has been done with recent effort, including producing polyploid plant by using somatic cell manipulation. Polyploidy of Artemisia annua using cultivation to obtain in vitro polyploid plantlet. Objective of this research was to investigate growth, stomata, and trichome characteristic of diploid and tetraploid accession of Artemisia annua grown in the field, and also is useful to evaluate the growth characteristic of polyploid Artemisia annua resulting from in vitro regeneration plant. The plant material used in this research that is KIB and K1 2.2 for a diploid plant and K 2.2.9 and K 3.2.1 for a tetraploid plant. Confirmation of polyploidy level using flow cytometer was conducted at Research Center for Biotechnology in the Indonesian Institute of Sciences, And also stomata and trichom measurement using Leica microscopy was conducted at Research Center for Biotechnology. Stomata and trichom measurement using scanning electron microscopy was conducted at PT Fajar Masmurni. Furthermore, for cultivation and plant growth measurement using seed produced from generation 1, planting in the field on the different bed, was conducted at Cibodas Botanic Garden. Artemisinin extraction using HPLC was conducted at Research Center for Chemistry, Indonesia Institute of Sciences, and the last for artemisinin determination for artemisinin content level was conducted at Center of Biomedical Applied Health Technology, National Institute of Health Research and Development, Ministry of Health, Republic Indonesia. Confirmation of ploidy level of fluorescence was being tested. The results show that KIB and Q1.2.2 were deployed and Q2.2.9 and Q3.2.1 were tetraploid. The peak size of deployed plant KIB was at channel 154.54. Q1.2.2 was on the channel 137.12. For the tetraploid, K2.2.9 on the channel 343.31 and K3.2.1 on the channel 300.78. For a stomata characteristic of diploid and tetraploid Artemisia annua plant also vary. The results show that abaxial epidermis cell had the highest stomata density than the adaxial cell. This is common for all plant species. Tetraploid Kit 2.2.9 had a largest size of stomata, so that stomata density was the lowest. The highest stomata density of adaxial cell was found at tetraploid Kit 2.2.9. However, the highest of the abaxial cells was found at diploid Q1.2.2. For a tetraploid Kit 2.2.9 clone, the size stomata was smaller than Q1.2.2 plant, which were also result of colchicine induction. It showed that the induction of polyploid with colchicine even took not doubling the number of chromosome. It affected the size of stomata. Trichome of Artemisia annua is placed for artemisinin and some other metabolite product. It from the data indicate that most abaxial cell had the higher number of trichome. Only diploid IB have a lower trichome number on their abaxial cell. However, diploid K1.2.2 and tetraploid K3.2.1 had a less number of trichome compared to that diploid KIB and tetraploid K2.2.9. The relative growth rate of Artemisia annua height was not significantly different. The relative growth rate of the number of the branches K3.2.1 was highest, different from the KIB, which had the lowest value. The biomass of the K3.2.1 tetraploid was the highest, but not significantly different from the accession tetraploid K2.2.9 and the diploid the K1.2.2. On the other hand, KIB, diploid accession has lowest biomass. Analysis of artemisinin of Artemisia annua by HPLC result that tetraploid plant had the highest artemisinin level compared to that diploid plant. The highest level of artemisinin was found on tetraploid K2 0.2.9. The lowest of the artemisinin level was on diploid Q1.2.2. For the conclusion, stomata, trichome, growth, and artemisinin content characteristic of Artemisia annua M0V0 was affected by the genomic of the plant. Growth of diploid and tetraploid plant were comparable, but the size of stomata and artemisinin content of tetraploid plant were more significant than of diploid plant. And the last, ploidy level was stable for both diploid and tetraploid plant. We would like to thank Darita Elvirantau for her assistance in stomata analysis. We also would like to thank PT Fajar Mas Murni for allowing us to use scanning electron microscopy. This research was funded by Center Biomedical and Applied Health Technology, National Institute of Health Research and Development, Ministry of Health Republic Indonesia, and Indonesian Institute of Sciences, year 2017 to 2019. Okay, thank you.
Thank you very much, Ibu Suluh, for the presentation. The next presenter is Bapak Sutomo with the title Krinyuk, Chromolaena adorata species distribution modeling and the potential use of fungal pathogens for its eradication.
The title of my presentation today is Krinyuk, Chromolaena odorata species distribution modeling and the potential use of fungal pathogens for its eradication. So Chromolaena odorata, or Krinyuk, or better known as cinnamon, is one of the weeds that is difficult to control and has causes many loss both economically and ecologically. This can cause ecologically negative impacts, a decrease in the amount of natural, native, or local biodiversity, as well as changes in ecosystem function. Species distribution model can identify areas at risk of further invasion by invasive alien species, so that early prevention of action can be done with appropriate approaches. Biocontrol should be less expensive, safe for the environment, and the species has to have suitable characteristics as a target for biocontrol agents. Some fungi are associated with Chromolaena odorata. However, the focus of attention to fungi as a biocontrol agent is still low. So, species occurrence data obtained from the Global Biodiversity Information Facility and combined with climate data from WorldCRIM, and then generalized linear model GLM approach was then applied to the data matrices. All analysis was done in the Biodiversity and Climate Change Virtual Laboratory, or BCCVL, application to obtain the species distribution model. So the method for biocontrol, the fungal isolation was according to Agrios 2005. Fungal identification was Plant Protection Biotechnology, Universitas Pajajaran, Bandung. So these are the results in discussion. So basically, Chromolaena odorata most likely to spread to eastern parts of Indonesia, although some parts of Java, especially in the south coast of East and Central Java, also predicted to be suitable for this species. Chromolaena odorata predicted to be suitable to inhabit Bali and Lombok Island with habitat suitability index approximately 0.8, as well as Lesser Sunda Islands, most of Flores and Sumba Islands, also suitable for Chromolaena odorata. Eastern Kalimantan with HSI 0.72, as well as Western Sulawesi and Papua are also predicted to be suitable for Chromolaena odorata with habitat suitability index approximately 0.84. And the fungal foliar disease with leaf spots was the most frequent symptoms occur on Chromolaena odorata. Chromolaena odorata plants commonly suffered from foliar disease with initial symptoms of necrosis and chlorosis. A total of 4 fungal isolates, namely from the genus Fusarium, Septoria, Poma, and Alternaria, were isolated, but all cultures has not been specifically identified. All cultures can be considered as fungal pathogens since they were isolated from the diseased leaf tissue. One species of fungus that can cause severe disease symptom on Chromolen odorata leaf was morphologically identified as Septoria species. This species can be useful for a biological control agent. Previous research reported that Septoria eggmaniana is a promising candidate for biological control of Chromolen odorata. Further studies, although all concerning disease intensity, pathogen identification, and pathogenicity, the tests are still needed to confirm the fungal status associated with Chromolen odorata. So we reach to the conclusion. With climate change to consider, Chromolen odorata most likely to spread to eastern parts of Indonesia. It is predicted to be suitable in Bali and Lombok Island, most of Flores and all Sumba Islands, eastern Kalimantan, and as well as western Sulawesi and Papua. One species of fungus that can cause severe disease symptom on Chromolen odorata leaves was morphologically identified as Septoria species. Further studies, though, are needed to confirm the fungal status associated with Chromolen odorata and to assess their biological control potential. And this ends my presentation today. Thank you very much. Good afternoon.
Thank you, Pak Sutomo, for the presentation. And last but not least, the presenter is Bapak Suyadi with the title Biodiversity in the Coastal Ecosystem of Small Islands and its Conservation Status.
Assalamualaikum warahmatullahi wabarakatuh. Hi everyone. Today I'm going to present our study. The title is Biodiversity in the Coastal Ecosystem of Small Islands and its Conservation Status. As we know that Indonesia is an archipelago country, we have more than 17,000 islands, and 77% of these islands are small islands. And we also the second longest coastline in the world. That means that Indonesia is a potential habitat for coastal biodiversity. And one of the biggest archipelago province in Indonesia is Maluku. We know that 92% of the area of this province is a sea, and 98% of the islands in this area are small islands. Indonesia also known as a mega biodiversity country, both for marine biodiversity and terrestrial biodiversity. For marine biodiversity, we can see here in the map that eastern part of Indonesia is the highest spot of marine biodiversity, especially in Maluku. And we identify some research care, for example, the coastal biodiversity, especially in the field, not only based on the model. The coastal biodiversity cover for the ecosystem biodiversity, species diversity, and genetic biodiversity. But in this study, we focus on ecosystem, especially mangrove, seagrass, and coral reef, and species, especially for mollusks, crabs, and fish. And we also assess the conservation status, especially the challenge of conservation and the opportunities of conservation. This data and information is required for management and sustainable use. So, in turn, they is important for conservation actions, and human well-being. The study area of our research is Ambon and Moa, located in Maluku, and this island situated in Banda Sea, Seram Sea, and Timor Sea. It's also known as the important Indonesian draw flow that connected in India Oceans and Pacific Ocean. This site also important for biodiversity and also important for regional and global climate regulation. Applied some methods, JS remote sensing and transects. We install some plots in transects, quadrats, and underwater photo transects, and we also did underwater visual senses along the transect. These methods to address the biodiversity and its conservation status. This is the results of our study. We found all marine ecosystem in the coastal, for example, mangrove, seagrass, and coral reef, is occurred in the coastal island, in the coastal of small islands. Let us start from mangrove. We find 15 species. We found 15 species of mangrove in Ambon and 5 species in Moa. And the diversity index also relatively high for 1.4 in Moa and 1.8 in Ambon. The conditions in Ambon, unfortunately, is not healthy or unhealthy or poor, and Moa is relatively healthy or moderate. How about the seagrass? The seagrass, we've found 10 species of seagrass in Moa and 6 species in Ambon. And diversity also higher in Moa than in Ambon. Different from seagrass, the coral reef, Ambon is much higher for coral reefs. And diversity index also is higher than in Moa. But the coverage is in Moa is higher than in Ambon. How about the marine biota associated with the ecosystem, for example, mollusks and crabs? For the mollusks, we found 100 species of mollusks in Moa and 30 species of mollusks in Ambon, and many of them are gastropods, and Moa diversity is higher than Ambon. And we can see here the similarity index in the graph or in the figure here. We can see that the similarity of mollusks between these two islands are less than 40%. This means that there is opportunity if we do more research to find more mollusks in this area. And for crab, also we find a lot of species, for example, 40 species in Moa and 25 species in Ambon. Interestingly, most of crabs in Moa is associated with seagrass, and in Ambon associated with mangrove. Moa diversity of crab is higher than Ambon. And again, the similarity of crab between these two islands is less than 25%. More study will find more species and maybe new species of both mollusks and crabs. How about the coral reef? We find a lot of number of coral reef species. For example, here we found 261 species, and the common species is Pterococaecia. We find in almost our study sites, but the most abundant species is Chromis tertatensis. And we also found the major fish 49%, target fish 42%, and less is indicator fish 9%. This means that the coral fish in the coastal ecosystem of small islands is not only important for economic values for markets, but also important for ecology, because indicator fish is important as an indicator for ecosystem health. And the diversity index also relatively high, ranging from 0.02 to 0.23. However, the biodiversity in the coastal ecosystem, they face some threats. For example, blast fishing, potassium, coral extraction, and bore or cartuba. We also find some challenge or threat. For example, if plastic pollution in Ambon is much higher, 1.68% of coral is covered by plastic, and in Moa 0.02%. Mangrove conversion into coastal development also occur, particularly in Ambon, and spatial competition of corals and sponges, sediment damage, and ulcerative white spots is the most common disease for the coral reef. And these threats or this challenge, it is predicted will be increased by 75%. And unfortunately, all of the biodiversity in our study area is located outside the protected area. But we still have hope because we have opportunity to conserve coastal biodiversity. For example, we have to recognize the local wisdom. For example, the Sasilau, and we have to do a public awareness to improve the knowledge, the perception of local people, so they to be more care to protect the biodiversity. And we also need to establish more coastal protected area, especially on the biodiversity hotspots in small islands and in the migration route. This is not only important to protect, but also to build a corridor of marine migration. In conclusion, we can conclude that small islands are important for coastal biodiversity spots because they have high biodiversity. They are important for many flexible species and location for migration routes for many species. And this coastal biodiversity also really depend on the local environmental conditions and threats in the small islands. The main threat, as I said before, is coastal ecosystem conversions, pollutions, biodiversity explosion, and sedimentation. So we need the new strategies. For example, one, we should establish a more coastal protected area in the small islands, build a marine corridor to protect migration routes. And we have to recognize local wisdom of Sasilau and public awareness for local communities. And we appreciate for Research Center for Deep Sea LIPI supporting this research, and we also thank for everyone who helped during the fieldwork and lab work. This is some reference. Thank you very much for listening.
Thank you, Pak Suyadi, for the presentation. And I think it is Pak Suyadi is the last presenters for this session. And we still have 15 minutes for the question and answer. And I think in the chat has already several questions, especially from Pak Sutomo. Maybe Bu Ruli can answer directly, and Bu Siti Sundari has already answered in the chat. Bu Ruliana, have you performed or have got spatial data of the change in vegetation density in your study area from certain period of up to now? This can be done by analyzing satellite image using NDVI, which will add more value to the research. 2. Dipterocarpaceae, which is the highest species richness, does it also has a good regeneration as well? For Bu Ruliana, please answer the question.
Okay, thank you very much. Thank you also to Pak Sutomo who already gave a question to my presentation. So actually, we haven't conducting the spatial analysis using the NDVI because at the time we are still trying to get more permanent plot to cover all the type of vegetations that we found there. So, and this is still one of the plot that we presented here, and the size is still 0.5 hectare. So at the beginning, we are planning to make a monitoring plot. So we are going to regularly monitor the plot. However, the fund of the project is already finished, so we are now still seeking for the continuations of the research. And for the second question, Dipterocarpaceae, which is the highest species richness, does it also has a good regeneration as well? We can say that even though it still need more time to conclude whether it is has a good regeneration or not, because as for the preliminary study, we can find Dipterocarpaceae species in many size of diameter. So from the small trees until the emerging trees. However, and also we found that the Dipterocarpaceae species has a mass flowering, but we didn't conducting the research of the viability of the seed and flower to reproduce into a seedling. That is, it will be good if we can continue the project in the future. Thank you very much. I hope this is answering your questions.
Yeah, thank you, Bu Ruli. I think that's the suggestion from Pak Sutomo is good to use for further analysis because it's like a satellite imagery, etc. It's very common now for research. And
Yes,
thank
indeed.
you, Bu Ruli and Pak Tomo for the question, and also from Pak Sunardi to Pak Sutomo. The question is, the picture of the leaf sample are more similar to Austropathurium inolifolium rather than Chromolena odorata, also the plant Statitus form in the first slide. Or other species has been verified from the herbarium. And the second question, masuk from Pak Suyadi to Pak Sutomo. What the parameter were applied to develop your spatial distribution or distribution maps? Thank you, Pak Suyati, for the question, and also please, Pak Sutomo, directly to answer the question.
Thank you, Mr. Cahyo. So, of course, there are several names for Crinue, but what we meant here is the Chromolena odorata. Previously, this species was named as Eupatorium odorata or Eupatorium odoratum, and yes, it has been identified at Bali Herbarium. And for the second questions, for creating the species distribution modeling map, we use several parameters, but mostly we use climatic data. We selected air temperature and rainfall variables as the variables for the environmental variable matrix for the species distribution model. Thank you.
Thank you, Pak Tomo, for the answers. And any other question or any participant want to raise hand for direct question for the presenters? Please, we still have a time, maybe 10 minutes, because we in a schedule we have up to 3:30. No, 3:20. So we still have about 10 minutes. Or anyone, please, if want to have a question to the presenters, or presenters want to comment other presenters.
Excuse me, Mr. Cahyo, can I ask question for Pak Sutomo?
Pak Suyadi, please, Pak Suyadi.
Okay, thank you very much, Pak Sutomo. This very nice presentations. I'm going to ask about your conclusions. You mentioned in your conclusion that climate change is considered about the spreading of C. odorata to eastern part of Indonesia. So, can you explain the detail and the reasons about the climate change? What parameter or how they work? So you assume or you find that this may be because of the climate change. Thank you.
Yes, thank you.
Pak Tomo, please.
Okay, species distribution model can predict the distribution model of a species in the future times. So beside the environmental parameters that are included in the model, I also included the climate change scenario, RCP, as a layers in the map. So it can actually calculate it for you and predict it 50 years in the future about how the species has shifted or changes in their habitat preference. So the model predicted that the Acromolena odorata will tends to change their preference from wetter climate area to drier climate area in the future. Thank you.
Thank you, Pak Tomo, for the answers. Any other question? And I will say thank you for Head of Research Center for Biology, Dr. Adit Kanti, and Head of Research Center for Biomaterial, Dr. Iman Hidayat, who attend this room, and we are grateful that Ibu Adit and Pak Iman can attend and can join this session. Hopefully, any question or suggestion for presenters or any comments? Please, Pak Iman or Ibu Atit.
No comment from me, Pak Cahyo.
Okay. Ibu Atit, any question, Bu Atit? Or comments for presenters before other participants? Maybe Bu Atit has other meetings. Any other question? We still have 5 minutes. If no more questions, I will give several announcements for all participants. First, for this session from 12 presenters, I will select one presenters for best presentation, and I have to propose to the committee. And the announcements for the best presenters will be announced in the Thursday on the closing ceremony on 20 of November. And hopefully all the participants in this room can join the closing ceremony. And we hope one of the presenters to be the best presenters during the international symposium. And then for the participants after this session requested to join the main room, so hope after the session finish, hopefully all the participants will join to the main room. I think that's announcement from the committee. And then all the presenters, thank you very much again for the great presentations, and also for all participants in the session. Also Pak Iman, once again, thank you very much for the time to join this meeting, and also Bu Atit and all the organizers who organized these meetings very well. And also, but not least, thank you very much for the video operators, Mr. Tulus, for very smooth presentation for the transition of during each presentation. And hopefully this symposium will be fruitful for the Indonesian biotechnology and bioengineering in the next futures. So hopefully all the result of this presentation will be useful for others subjects. Thank you very much, and apologize for any errors during the sessions. And once again, hopefully all of you enjoy the symposium. Thank you very much. Terima kasih Pak Iman.
Sama-sama, sukses. Tapi presentasinya bagus-bagus.
Okay.
Thank you very much, Kak Cahyo, as moderator, and everybody for the presenter and participants.
Okay, everyone. Thank you very much, and good luck for the best
For
presentation.
all participants, please start the video because we will take a screenshot for the photo together.
Oh,
please. Everyone ready? So all participants, please open your camera. So we will start. 1, 2, 3. Wait a moment. And one more. 1, 2, 3. Thank you, and see you in the main room. For all participants can back to the main room by leaving the break room 1. Thank you.
Ladies and gentlemen, we have followed all agenda today. Thank you very much for all speakers, moderators, distinguished guests, and participants who have attended and actively participated in the symposium. Tomorrow, we will continue our symposium. To all participants, please be aware to change your username according to the format given in the conference room. Please join on time. Thank you very much and see you in the next session.